Is phone in the position? It might be on the podium. Yes. I think I'll have to start without them. It's 9:35. My own boys, where are they? Yeah, they have come. Yes, they are here. Let's lock this. That's a good idea. That back one is enough. Yes. Yeah, my boys are here, so let's start. I want to introduce you to Vivian David Jacob also. Hi, Vivian. So, Vivian did a master's course in VIT. He did his project also with me. So, the way Vivian and I started off was that, in these elective courses, there needs to be, but you guys can sit here. I don't think we are going to have any VIPs and all that. You guys can sit here. If you want, you can come here and sit. I'll feel better that way. My guys in the front. So nice. Okay. Come on, guys. Why don't you come here? Here. See here. Please come here. So it doesn't look good. The front seats are empty and the back seats are taken. Speaks poorly. Yes or no? Yes. Now it's okay. Good morning to you. Thank you for your presence and participation. So today we'll start at around 9:30. I had thought of 9. And so the session is going to be packed because this part is filled with experimental data. And that's what people like Karthi like. He appreciated those slides where there was data. Scientists like data because the other aspects are mere interpretations by people. Data, they can themselves sit and make for what it is, right? So scientists dig data. That means they find data and they love data. That dig word has two meanings. Okay. Okay, thank you. So let me give you a brief overview of what we have done till now. First session was a snapshot of what murburn concept is, how it explains the role of oxygen in life, and what are the different fields that it has impacted. Just a bird's eye view, all right? And then, if you could come here, my span length would be reduced. If you can take that seat, please, thank you so much. So that I can only be bothered about this part. Yeah, that will be wonderful. Yeah, no, it's okay. It's okay. Now it's okay. No, wonderful. So, thanks for cooperating. So, that was the first session. And then, earlier part itself, you were informed that this work is very important. So, it cannot be something that you just waltz in and waltz out at your own jolly will. The raison d'être of biological pursuit. What is life? The quintessence of life. All right. Now, in the second session, what did we have? We had the data, the gory details of how murburn concept was established in which system? She said it. Which system was murburn system, Murzyme first established? Chloroperoxidase. Okay. What is chloroperoxidase? Feel free to speak. It's a protein, of course. It's a protein. That's a good answer. What's Murali? A living being. Yes, good answer. But more specifically, chloroperoxidase is a fungal heme enzyme. Yes or no? Yes. So it was an extracellular enzyme. Then we came to another couple of examples. What were they? Wonderful. Hemoglobin and lactate dehydrogenase. Hemoglobin and lactate dehydrogenase. We found new function for hemoglobin, whereas lactate dehydrogenase, we disestablished certain ideas. What was the most important idea about lactate dehydrogenase that we de-established? Yes, exactly. It is not a free for all. You can go this way, you can go that way. No, it doesn't happen. Enzymes are not miracle workers. What do enzymes do? Enzymes allow, the most important concept that you learnt is what? One word, attain equilibrium. That's what they do. They can't mess with that, right? And that is the law of thermodynamics. All right, everything governs that. Okay, now we are going into more complex systems. In the earlier systems, the first chloroperoxidase, it was a single enzyme, single protein, right? And this lactate dehydrogenase and hemoglobin, they were multimeric. Some amount of complexity, but not the kind of complexity that you're going to see today. In the first session, you're going to see drug metabolism. In the second session, drug metabolism, where does it happen? Primarily, liver. Okay? Everybody appreciates the mechanical heart because it pumps the blood, brain because it processes information, and kidney sometimes, what does it do? Filter things. But the metabolic mainstay of your body is liver. It is a sentinel organ in your system. What is a sentinel? Sentinel is somebody who guards the place. So you have a mouth, you have an anus, and whatever goes through is a tube. All right, that's not theoretically within your body. Your body is theoretically the space between that alimentary canal and your skin. That's where you have to regulate things. You can take whatever nonsense into your system. It will chuck out. But whatever is taken in is first processed by liver. So that system, how does the liver look? That's look, very good. The contour you show, that's one answer. It looks reddish brown. One thing we explained yesterday, I forgot. One message we forgot to give. What was it? Yesterday, when we gave the overview, we had said that the prevailing ideas could not explain how the red blood cells are viable for 120 days without nucleus and without mitochondria. Part of the answer was afforded yesterday. Yes or no? Why? Because if ATP is required, the amount of ATP that is required is far below what can be given by glycolytic intermediates, because the glycolytic intermediates are at much lower level. By law of mass action, you cannot get it there. Okay, and ATP is not something that is going to sit there happily. It is going to that's good, nice. Now those of you who are sitting through all the sessions, we'll take attendance, and only if you meet the attendance requirement, we will if you don't meet the attendance requirement, we'll ask you to pay for your lunch and dinner and the accommodation, because you should be here primarily for the symposium. You're supposed to be here. Majority of you are students, and students have to be controlled. There's no freedom for you. Those faculty and students, faculty and scholars, I won't control, but students need to be controlled. I strongly believe in that. There's no concept of freedom for them. So what was I saying? The mitochondria. Without mitochondria, how can the erythrocytes or RBC be viable for such a long time? All right. So why? the energy requirement is taken care by murburn reaction, sponsored by the tetrameric hemoglobin. We found why the monomer exists, why the beta. It was just sitting there. No role was given to it. All right. And lactate dehydrogenase, why was the Cori cycle? Why should lactate that is formed in the muscles when you run or exert at a stretch, the lactic acid that is formed in your muscles, even though they have 4 times higher LDHA than the same LDHA that is present in the liver, it is still going to liver because liver is the fundamental place of murburn. So liver is the place where I coined the term murburn. It is the center of murburn activity, if you call, that is the reddish brown. Can you make a note? Blood is also red. This is also red. There are some other colored things because of heme. Fundamentally, heme is a very big cofactor in your system, and that has been present from the day 1 of evolution till now. Only for what? No, what this context. The insight. Yes, it is a murzyme. Heme is a cofactor in multiple proteins, right? So with that introduction, can I go on to today's talk? I have given you a context. So today's first talk, yesterday all the enzymes were based in water. They were aqueous systems. Now we have got interfacial systems. We have got lipid embedded systems. Okay. So I think this, okay, this slide is okay. So good. Will the video be better if I stand here, or it's okay? No, the video I was asking them, because this is for posterity. Yeah, this is recording. I think he has put it here. That's better. So I'll come into the face like this. This is better. So, but if I do that, I think this is a better position, I think. So, yes. So, now we studied chloroperoxidase system, which is a heme thiolate enzyme. And if you see cytochrome P450, it is also a heme enzyme. It is also a heme thiolate. It has a cysteine amino acid residue that is ligated to the iron, and it is also around approximately 45 kD, same as chloroperoxidase. So it also can do a wide variety of reactions. Do you remember it? I said that chloroperoxidase can do multiple types of oxygen insertion. Cytochrome P50 also does oxygen insertion. So here it also does activated C and heteroatom oxidation. So dealkylation, demethylation, NS oxidations, all that it does. So the other similarities are, there are some differences. However, fungal and extracellular are highly glycosylated, polar active sites, whereas the liver microsomal are membrane bound and they lack major modifications, and they have a hydrophobic active site. Active site is what? The heme enzyme pocket. Okay. Now, so this can also do chlorination, whereas this cannot do chlorination. This requires reductase, NADPH, and oxygen and cytochrome B5, whereas this has hydroperoxide. So there are differences in the reaction system. But if you understand a bit of chemistry, all these superficial differences that biologists see, as a chemist or as a physical chemist, you will understand that that's not a major deal. You try to understand a biological reaction or a biological process for the chemico-physical aspect of it, and don't see merely the distinction based on some this component, that component, like that. What you start to follow is, where is the electron coming from? Where is the electron going to? Or what moiety is transferred from where? What is it going to? And if it is a macroscopic phenomena, how could the phase change? things like that. That is what phenomena or a process is fundamentally about. Okay, so that's what we'll be dealing with now here. So this is responsible, chloroperoxidase, that I dealt with in great detail yesterday. It is responsible for halogenated molecules in environment. And have you heard the term xenobiotics? What is xenobiotic? What is a xenobiotic? Can anybody give me an example of a xenobiotic and differentiate it from a non-xenobiotic? Anybody? One xenobiotic molecule. It's a foreign compound. Okay. A non-xenobiotic, don't murmur it within you, speak out. Only when you speak out, I repeatedly say, your confidence goes higher. If you sit at the back, murmur it, you can try to tell yourself that I know this, I know that. But then the merit as a scientist will come only if you claim something. You say, this is what is new. You have to go out and say it. If you just sit at the back or try to be non-expressive, non-communicative, it's not going to help you as a scientist. So that's what my primary objective is. My primary objective is not to give you a lot of information. All my work is already published. What primary objective is to get you to interact and enjoy the process of doing science. And the process of doing science has to put your ego into perspective. Your I should come into perspective. Did you get that message very clearly yesterday? If you did not get it now. So, xenobiotic is something that is not actually a part of your evolutionary history. It's something that is completely alien. You know, your system cannot recognize it as something that it has to process. It looks a crazy molecule. Suppose I know Vivian. I can make out what Vivian is because I am familiar with his topography, facial features, body, all that, dimensions and all that. But what now you eat food, right? Food is composed of what major three polymers? Carbohydrates, proteins, fats. So these molecules, you have the enzymes that beautifully cut, deterministically cut, chop, break it into smaller bits, that digestive process. That's wonderful. They are a part of evolution. But suppose a pesticide gets into your body. All right? Does your system know how? No, it doesn't know. So how fundamentally a molecule, don't you think such molecules can get into your system? Because your system is not somebody like, do you think your country is always regulated by passport checking people at airports? You have such a huge boundary that is unmanned. If there are people who can go, there can be animals, they can just come in and go. So like that, similarly, like a country, the cell membrane or cell wall is not something that can completely safeguard whatever you want, whatever you don't want. So that part is what we are dealing with. Xenobiotics is molecules which are not required, which are not familiar to your metabolic system. All right. So what does the liver metabolic system fundamentally comprise of? Now, in all these systems that you saw yesterday, it was one simple enzyme and then one or two substrate. And then what I did was I put an extra something like azide or something like cyanide or some molecule to see what was how the system reported. How do biochemists find a mechanism? Biochemists find a mechanism by putting some molecule inside and seeing the effect. Do you understand? So now, is it a closed system? If I want to find, how do I do? I'll check. I'll put something in. Am I seeing that molecule coming out from some other aperture? Something like that. I'll trace like that, right? So similarly, I can understand a biochemical process better only if I have a way of finding if something happens properly, wonderfully, then I put something in, it doesn't happen. So that says something important about that procedure, what happens. So that inhibitions are a important way of understanding an enzyme mechanism. So the cyanide and azide that I put in heme enzyme system, and I tried to do some, we reported some data yesterday, was something like that to understand the mechanism, how the substrate and enzyme interact in that system. Here you have NADPH, which is an electron rich molecule. Here is a nucleotide part, and here is a nicotinamide part, and then you have one diflavoenzyme called reductase. I shall call it CPR. What is CPR? Cytochrome P450 reductase, and this is cytochrome P450. Cytochrome P450 is a heme enzyme. This is a flavin enzyme. Okay, you know how flavin looks? Flavin is a 3 nucleus organic compound. Okay, now it has got nitrogen hetero atom inside in the nucleus. Okay, so this is nicotinamide adenine dinucleotide phosphate reduced. Then this is the 2 enzymes. Then it uses molecular oxygen, and some enzyme systems also use cytochrome B5. This is a membrane bound enzyme. This is a membrane bound enzyme. This is a membrane bound enzyme. This is a water soluble molecule which can go into the interface, and this is a gas that is freely diffusible. So these are the fundamental nature of the reaction system. When you are a biologist trying to understand a process, you have to understand what are the components of the process, and only then you can understand what is the mechanism. All right. So now now what is the importance of this? What is the importance of this? All the drugs that you consume, are they xenobiotics? Yeah. Majority, vast majority. Is there water here? Yeah. Yeah. This guy wanted to be desperately attending the conference. No, thank you. And now you see him. You give a man a little space, what he does. Yeah. Right. I told you, Prasad, he was putting up a show. He was, wasn't he? It's not good for you. You're not creating a good impression. How does science work? Let me tell you how science works. Science works by, like all ways, 3 ways you have to succeed in life. That is more important. Let me tell you that. This kind of things put me to a, digressed path, a divert pathway. I feel very bad when young people get opportunity and they don't use it. So you as a youngster, you need good habits, good skills, and good relations. And when you come to symposia like this, you network with somebody, and you meet somebody, and you give a favorable impression. And that favorable impression carries a long way. Now, I know Niranjana. She's sitting there. She answered. She is coming here. I know. I've seen. I know Muhammad is doing that. I know he is doing that. I forgot your name. Muhammad, right? Karthik. Sorry. Karthik is doing that. So, Priyam is also being attentive. Muhammad is also being attentive. So, I know now slowly. I'm a powerful guy in the future. Who would I advance in their life? Who would I? People who goof around or people who mean stuff? I'll support people who mean stuff. There's no point in grinning, my boy. There's no point in grinning. When you have to be serious, you have to be serious. When you are serious, you can be funny. When you are all funny, you cannot get serious. Because potential is developed, you can dissipate that potential very easily. Developing that potential requires a long amount of training, sustained practice. All right? So you see, the drug molecules, what is that industry? How big is that industry? $1 trillion industry. The Czechoslovakian premier just said something about COVID. What happened to him? Nothing political here. But you see, drug pharma industry, very strong lobby. All right. So what we are doing is very, very important today. So how do the diverse drugs that get into your body that you're administered? What if you take a tablet of aspirin and it is says 100 milligram, 200 milligram, whatever it is, some molecule you take in, your body doesn't metabolize. Then Abhinav takes in 10 tablets, and Abhinav will not be Abhinav. Abhinav will be aspirin plus Abhinav, right? It doesn't go out of Abhinav. It has to go out, right? That thing has to be done by this guy, this enzyme system. And this is one of the most thoroughly researched field. So why is it called a cytochrome P450? Cytochrome P450 because this heme thiolate system, when it is reduced, how is it reduced? It electron giving chemical ion called dicyanate is added. It is a historical experimental procedure. And if you put carbon monoxide, we spoke about what and all molecules yesterday: oxygen, cyanide, azide. So carbon monoxide is yet another thing that binds with heme. So carbon monoxide, if you put, you get a characteristic spectrum that peaks at 450 nanometer. The heme histidylate systems like hemoglobin and all that, they don't give that 450. They give around 420. All right. So that's the difference why it's called cytochrome P450. So this will be called CYP from now on in this talk. All right. So it gives, since we are talking about active site processes, we will talk about binding, because binding is the classical way of explaining enzyme reaction at the active site. So there are two types of active site bindings. One is the type 1 as shown by P450 CAM. P450 CAM is a, have you heard of the bacterium Pseudomonas putida? Yes. So camphor is a hydrocarbon. This Pseudomonas putida, it has got a cytochrome P450 which can metabolize camphor very effectively. So those of you who do environmental studies, this is directly related to hydrocarbon, remediation and all that. So that kind of bacteria uses this cytochrome P450. Otherwise, cytochrome P450 are not easily found in that high amounts in bacteria, all that. So here you have type 1 where the substrate goes and binds here, but it does not directly chelate with the heme ion, and there is then the heterocyclic nitrogen containing kind of metyrapone, for example. It goes directly, the nitrogen chelates with the central ion. So there are two types of binding. This kind of an important information is required because it tells you something about cytochrome P450, some ways of studying cytochrome P450. So now, before I went to, I went back to US. I came after 2000. I came back. I did social work, and then I tried to get into IIT as a faculty, and I did not because I wore a dhoti and kurta to the interview, and they did not like it. So I was outspoken. Some faculty said, because we have had long history of argumentative discussions. So, and I'm always argumentative. I hope scientists, if you are planning, you need to be discussing, arguing. So here is the theory that was prevailing in the lab. I went to the Washington State University, Professor Jeff Jones lab. All right. Very nice man. Personally, I liked him a lot. But when I differed with him on the mechanistic cycle, I was thrown out. All right. But he gave me time to work and finish it. But when I reported my findings to him, he said, you cannot shake the fundamental hypothesis in the field. That's what he said. That's so sad. That's not what a scientist does. That's not what a mechanistic scientist does. So Jeff should listen to this, and there's no running away from this. So this was a theory that he was using, and all the other people in the world use even now. All right, even though they know my work, they continue to apply this old theory. What is the theory? Heme activates oxygen. That is, because hemoglobin binds and moves oxygen, cytochrome oxidase is supposed to bind oxygen, all heme is supposed to bind oxygen, and then cytochrome P50 reductase, once, there are two proteins right here. What are the two proteins? Cytochrome P50 reductase, which is a flavoenzyme, cytochrome P50, which is a heme enzyme. Now, there is something called redox potential. You know what is redox potential? Yeah, it's a measure of the level until which how strong an oxidizing agent or how strong a reducing agent is. So when you see the cytochrome P450, its heme ion is around at minus 350 millivolt generally, minus 350 millivolt. But when you see reductase, its redox potential, flavine's redox potential is around minus 250, minus 270. So electron will go from reductase to cytochrome P50, or in that state, minus 350 to minus 250, will the electron come, or minus 250 to minus 350, minus 280 or whatever? Higher to lower, that is not an answer. Minus 350 millivolts to minus 250 millivolts. My question was very specific because we are dealing with negative here. Higher, lower is not something that you can easily relate like that. I have asked a very specific question. Will the electron in normal state, cytochrome P450 is minus 350. Will the electron go from cytochrome P450 to reductase, which is minus 280 or something? Will it go like that, or from reductase to cytochrome P50? Don't do this. You have to speak loud. By doing this, you are convincing yourself that you are communicating. You are showing me that you are communicating, but you are fooling yourself. You are not fooling me. Do you understand what I'm saying? I want you to go out. Speak whatever it is. It doesn't matter. Speak what you are saying. Don't see there. Then you are thinking what the others are thinking. You're not doing anything shameful. You're doing the primary activity of science. Go ahead. Wonderful. What's your name, girl? Indumati. Good, Indumati. The electron, it's a fundamental concept. Minus 350 to minus 250. Because the electron will always go to positive, right? It will like very simple ideas. If you have such simple ideas, you can understand and build everything yourself. So, but then, what does the classical mechanistic cycle require? It requires that reductase should give electron to cytochrome P50. So, they found in cytochrome P50 CAM, putida, that thing, that the camphor binds to cytochrome P450 and somehow it lowers the redox, it enhances the redox potential. That is, it increases the redox potential from minus 350 to say minus 350 to minus 250 or something. So minus 280 is flavin, then it can receive the electron, right? So the presence of the substrate acts as a switch. Did you understand? Only when the substrate is present, the reductase goes and gives the electron. Once the heme is reduced, it can bind oxygen. Yes? And then the oxygen gets further activated at the heme center to make same compound one that was in chloroperoxidase also. You understand? Porphyrin pi-cation radical that was produced in the enzyme active site of chloroperoxidase. That same compound one is also there in cytochrome oxidase, supposed to be, that is responsible for in your mitochondria. There are similarities in all. So something that becomes popular or accepted in something becomes the norm in all the fields. So here this is a very complex enzyme cycle. Here you have the substrate is required, then the reductase is required to bind to NADPH and get the electrons, and then it is supposed to bind to this. So four or five molecules are supposed to come in a particular sequence and form protein small molecule interactions and very deterministically go through an active site process. Here diffusible reactive species like peroxide are known to be produced in the milieu, but they are produced at the heme ion. So, and there is, suppose I use 5 molecule of NADPH, sometimes I should ideally get 5 molecules of substrate hydroxylated. That is the stoichiometry. But sometimes I get only 3. They say that water is formed here, and the loss of water is because of water shunt at the heme. So peroxide, superoxide, loss of redox equivalents from NADPH is explained by the chemistry at the heme center. So you see what the mechanism was before I got into the lab. So first day I saw this, I took a printout of this, I put a cross on it within the first week itself, and I put it in my table. My PA gets upset. I thought that this was highly improbable. Why? Even if the world believes in, my basic understanding of chemistry told me that such a diverse number of molecules cannot come deterministically in a sequence of events and go about doing this. All right. So here you see the substrate must be wedded at the beginning and it must get hydroxylated and come out. So now you take a molecule. What is your system basically comprised of? Your system is basically hydrophobic. Your cell membrane is what? Lipid. It is hydrophobic. So the hydrophobic lipid is what is, the way to get into the cell. When all these xenobiotics, they get into the membrane, they will get stuck there. So this hydroxylation, what does it do? It makes the molecule polar. And then the water can wash it. And then the kidney can pass it out. That's the logic. Alright. So the classical enzyme mechanism is through an active site, systematic, deterministic, multi-protein, sequential mechanism. Now what did I say? The murburn model says it is not the heme that activates oxygen, it is the flavin that activates oxygen. Reductase produces a diffusible species that can get into equilibrium, and this diffusible species is used by cytochrome P450, and the substrate needs to be bound only transiently or be in the vicinity, and it can make that. And then this ROS, reactive oxygen species, can react among themselves and give water. That is explaining the loss of redox equivalents. This is an alternative theory. So I'll quickly go into multiple views of the same theory. Do you want me to take a break and try to tell you something else? Or are you with me? With you. Okay. So basically the class, yes. Okay. I will show you that the same difference in multiple renditions. The same mechanistic difference. I hope you will get that. What is the classical proposal and what is the murburn proposal? So the classical proposal is first cytochrome P50 heme enzyme binds to the substrate, then reductase binds to these two bimolecular complex, then oxygen gets binds. So you have 1, 2, 3, 4, and then even cytochrome B5 can also come in, and then it goes through a cascade of events. Whereas this is two parallel enzyme systems working independently. They don't need to bind together. And all molecules just need to come transiently together. They don't need to bind, stay wedded for a lifetime and, lived happily ever after kind of thing, prince and princess and making like that. So here is yet another rendition of that. That is low spin cytochrome P50 becomes high spin, that is the redox potential changes, and then the NADPH binds and gives the electrons to reductase. They give multimolecular complexation and in transfer, and then electron gets pumped from the reductase to the P450, and then oxygen binds, and then you have the catalytic cycle. And the loss, and then cytochrome B5 can also take electrons from here and there to give the protein outcome. Here, the new theory says oxygen interacts and oxygen diffusible reactive species come out, and then this is getting into equilibrium with the milieu, and that is used by cytochrome P50 and produces the product. So yet another rendition of that. That is, here you have the classical substrate, switch on, oxygen, then intermediate steps. The coupled reaction gives the ROH, the uncoupled gives reactive oxygen species. So in the classical model, reactive oxygen species is a waste, something which leads to loss of redox equivalent from NADPH. NADPH is what? Giving the electrons. You should understand how this is. It's a electrons come, something gets reacted, that changes its property, and then it is taken out. All right? Now, my theory is oxygen is activated by reductase. Ross, not my theory, the murburn model. There are many people, I should stop doing this now. There are many people who have worked on it. It is murburn. You see, I'm hypercritical about myself. When I critique you, others, I am critical about myself also. So it's not critical approach towards anything is not bad, as long as it is done transparently, honestly, and sincerely. All right? Always remember that. It is not done with an ulterior motive. I do not get anything by admonishing a younger fellow. I know that I have gone through that age. Perhaps at you people's age, I was also like that. Perhaps. That's yet I will try to, just because babies go potty in their own shorts, we try to discourage that. That is the whole point, right? Bad habits have to go away. So the newly proposed one, substrate required in the one step, last step only, no protein complexation, substrate hydrolysate needs ROS. So my project with Jeff Jones was to minimize ROS. That is what the pipeline of drug optimization in the pharma industry, which goes through 14, 15 years, 10 to 15 years, goes through. There are several lead molecules, only some become the drug candidates, and they have to go through such optimization procedure. One major thing is they should not produce ROS. I reported to my PI, if you take away the ROS, there is no reaction. The fundamental thesis, the fundamental hypothesis in the field is flawed. That was my take. He said, you can't do it. So, yet another way to differentiate the theory. That is, the substrate comes and binds. There is it's bound at a different place. There is an active species. Then the substrate gets to be within bonding distance of the interactive species, and then there is OH formation and the OH goes out. Substrate binds, compound bond forms, then substrate is at bonding distance, dissociation of the product. So I have shown you the classical, and here what is the difference? It's a very subtle difference, but it's a very salient difference. So have you heard of this joke? Somebody said that there were three guys, one guy said, our planes go out, practically go to outer space and come back. The Indian asked, my goodness, is that true? No, some few kilometers below the end of this thing, Okay. The Russian said, well, we guys build submarines that can kiss Marina Trench and come back. So, oh really, can it withstand that? No, some kilometers ago. Like, Indian said, we in India eat through nose. Huh? Really? No, it's just 1 centimeter. All right, 1 centimeter. That minute difference, goody difference, very, very small difference. One difference. What is the difference? Here in the murburn model, the enzyme does not require, the mechanism does not require the molecule to get into the heme pocket. Heme pocket is deep rooted. I showed you chloroperoxidase structure. Did I show yesterday? There was a channel leading into the active site. So the heme forms a floor. The molecule has to come in. So all the molecules need not go there. So there is a reactive oxygen species that is stabilized here, and then it goes out. Molecules of different topography can be bound here and there, and they can get reacted. This is a very small difference, but very important difference. So this, a molecule can combine here, there. If while the ROS, when it comes out, if it can react, you can explain the catalytic outcome. Okay. So how did I show that with the experimental data? That is what is the next aspect. So the tea is going to come in any time soon. 11 o'clock this session ends. So I have to be a little quicker now. So here you see, what is this thesis? Here is where the exploration part comes in. What did I do in chloroperoxidase reaction? Remember what was the first thing I did? Saw whether peroxide actually stops the reaction. Here what did I do? Okay, if their theories explore, resolve, prioritize. First principle of my philosophy. Before forming hypothesis, explore, see what does what. Is it heme that is activating? Is it flavin? Just two things mixing. You don't know what does what, right? So to ascertain for myself, I put some reactions wherein I get commercial or pure enzyme systems, and I mix myself, and I try to trace the reactive oxygen species without and with substrate. Without is this one, the filled black. With is the empty one. So wherein do you think is the peroxide concentration higher? Can you tell me? Which systems are the peroxide concentrations higher? With substrate or without substrate? Without substrate. Good. So without substrate. So what does it do to the theory? Huh? What does it do to the theory? It contradicts. The theory should be jettisoned. What did Caroline Wells say? Remember my first slide. Boom. Discard. So because this doesn't agree with it. So both reconstituted pure peroxide and milieu over time. So in peroxide also changed in time. When you give peroxide to heme, there is no reaction in cytochrome P450. Whereas in chloroperoxidase, it converts. Here, it's a hydrophobic pocket. It's a one small change. Hydrophobic pocket cannot process peroxide. So, hydrophobic pocket can deal with superoxide. All right. So, that's what is the difference. Here you see that peroxide is changing in time in different systems. You can see that is the realistic. What did Tycho Brahe do? He got data points, but he did not see that it was ellipse. Kepler put that data points to the right theory. Yes? Now, people would see this and they would say, no, these things are not important. For me, explaining natural phenomena was more important than fitting some data points and disregarding some data points. Remember that ant circle, that thing? Always remember. Good approaches, good cardinal principles. Science is not a matter for very smart people. I would say science is for very bull-headed people. They believe in certain things, certain way of doing things, and that's how they do it. Einstein and all were great geniuses. I don't consider myself a genius. Just bull-headed. Simple fundas, simple things, that's it. And that's how you can do science. And be confident about it. Go ahead. So that's the kind. So the next thing is, so first thing it clearly, their theory didn't hold water. Okay, the mechanism cycle was not good. Then the next thing is, see here I have used multiple isozymes, pure proteins and all that. In taking the global data space, not just working with one, exploring adequately. That is very important. All right, not just going by some kind of consensus. Here, the reaction system, they always added superoxide dismutase and catalase. What do these enzymes do? Anybody knows? They are antioxidant enzymes. Basically, if my theory that cytochrome P50 uses ROS for the catalytic cycle is true, then SOD and catalase should take away the peroxide and superoxide, and I should not get any hydroxylation, right, of the substrate. But then the hydroxylation is still efficient, like the control. This is the control. This is the loss of NADPH, rate of loss of NADPH. This is the product formation of hydroxylation. So this is a substrate. This is a substrate depletion rate. This is a product formed. Now the point. Now you see, when I add ROS utilizing enzymes, it doesn't go away. The product is formed, and in fact, it's very efficient. Very less amount of the redox equivalents are used. But you see, another enzyme which is somewhat hydrophobic can highly deplete the ROS, because this can use superoxide, peroxide, all that, and there is no product formation. Fluoroperoxidase, which is inactive at pH 7, doesn't affect the reaction much at all. Now, this would say that ROS is not involved in the catalytic mechanism. Yes or no? That is what the data says. But probing the data further with HRP showed that the ROS is getting manipulated, NADPH is getting affected, with reductase also, and we just did a very, very simple experiment. What is ascorbate? Anybody? What is ascorbate? Vitamin C. You get a chocolate, sir. From now on, you cannot answer my question until and unless. Yes. Yes. Yeah. No. So why are you sorry? No, it's just the rule of the game. That's all. You don't need to be sorry. So because I have only 15 odd chocolates, huh? That's the reason. Otherwise, I could have given you chocolate and you would have had diabetes or something. No, I'm just kidding. So after some time, you have to unwind so that some people who are sleeping can get back to, yeah. So here is ascorbate, that is vitamin C. What is vitamin C? Is it a polar molecule or a hydrophobic molecule? Polar says polar. Yeah, and you see the structure. It's a polar one. Here was 50-50 chance. Polar, non-polar. What do you get a chocolate? Saying it clearly without merging into the background, saying it as an independent person. That's more important. I am encouraging that. For me, all these details are secondary. For me, this is society building. This is what I'm interested in. Do you appreciate that? You understand that? Yes. Okay. So here, what I did is, I took ascorbate and I added, like what SOD or catalase was doing, some marginal enhancement. But I put a fatty acyl substitution to that ascorbate. One aside, this palmitate, this is a long chain. See, you see, same molecule, but it has got a derivatization. So it will be localized in the membrane where cytochrome P50 reductase are placed. Now what happens? Hydroxylation gone. What does it say about the mechanism? Here you got an enzyme that can take the ROS in hydrophobic areas. Here you have got These are soluble enzymes. So the access, the acquiring of the reactive oxygen species in the site where the production and utilization is, that is crucial. What this does is affect the Ross equilibrium in aqueous phase. This can alter it into the lipid phase. So if you remember my slides, I said delta G of formation, partitioning effects, all that. So you see how murburn, why this shows you why we have lipid membranes. Oxygen is more soluble in lipids than in water. That's why you don't have a protein cell membrane or a carbohydrate cell membrane. You have lipid. So murburn is what explains the core basic competence of components of all fundamental activity. So now so in that case, if I take cytochrome P450 and add reactive oxygen species like superoxide, 1 electron reactive oxygen, I should get the reaction, right? It's a very simple thing. I did that, I got it. For different sips, different reactions, I got it, the control reaction. Then, in that case, if I separate the reductase and cytochrome P50 by a dialysis membrane, I should get the reaction. Yes, because the soluble species can go through dialysis membrane, right? I get it. There is the specific production, but very low, because the reactive oxygen species are very transient. So the question that you asked yesterday, how can I accept when all my life I have learned that reactive oxygen species are basically not good? Well, reactive oxygen species are being produced and used very fast, very quickly at the place where they're supposed to be used. When they don't get used in a very efficient manner, they get accumulated here and there, then you have a problem. Now, if you're working with an engine, if you see oil dripping from some place and then you say there's something problem, you don't say that oil is the problem. Do you say? Huh? But only in this case, we have interpretations like that. Do you understand? Understood? So, I have to give you such explanations so that it goes into analogy. Analogy helps us understand, no? Sometimes all these chemical structures, they irritate us. But then I have learned to love molecules for their structures. For me, this is like seeing Vivian or something like that. For me, looking at the molecules, that I told you that after that master's level, that exam thingy, I did not really like those molecules, and everything was, slowly I started to appreciate what molecules look like. I started to see them in a three-dimensional space. I started to visualize how they go into it, and I started making difference between elephants in Maruti and the other explanation. Did you understand that? That process of coming to the truth has to be grappling with the idea of reality. So now the loss of redox equivalence is more by reductase mediated reaction. So when I took peroxide and I conducted the reaction, it is facilitated by the flavin enzyme. So loss of redox equivalence is more because of reductase mediated process, not at the heme enzyme. So like that. So confirmed it in multiple ways with some other experiments. So basically this part is to understand the reaction system. Okay. So here, do you want to take on here, or do you want me to go ahead with it? No, as you wish. Aap chahte ho? Okay. Okay, you can. Okay. So now, here is what the certain experimental aspects I had done. Then I was fired from US, right? 3 labs fired me for my ideas. I said, what the hell, land of opportunities? Jao. I came to India. I established a lab. I got my students. And my students explored it with whatever little facilities we had. And I told you, this guy got to be the receiving, but his thesis was held up for having that. Yeah. So now is his moment. All those people who blocked us and all that, I'm not going to be very magnanimous and all that. No, 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 no. No magnanimity. No magnanimity. People are supposed to be open-minded. Right? Managements will not like me being like this. Because for them, it should be smooth. But then, managements are supposed to be smart also. You should be able to differentiate. When you are sitting, society is giving you so much prestige, so much comfort, respect in society. What is it for? It is for knowing things, being open-minded, not being smooth, whatever everybody says. No. So, Abhinav, this is your moment. Go ahead.
Am I audible? Okay. So, no, that's okay.
No. I will go back.
Yeah. So, all this time we have seen this P450 system, SIP system, and systematically sir has shown that the oxygen activation or the reaction is not happening through the classical understanding that has been. What has been the classical understanding? The substrate has to come and bind to the active site, okay? And obviously when the substrate comes and binds to the active site, you need a channel from where the substrate has to come inside, right? And then
one setup பண்ணிக்கலாம். Wonderful. கொஞ்சம் நேரத்துக்குள்ள வந்து. Thanks. ஒரு அரை மணி நேரம் வந்து.
சந்திரன் பேசணும்.
யாரு?
சந்திரன். Corporation. Connected to the mic.
Oh, okay.
Okay, yeah. So let's come back to the reaction system. Now we have seen cytochrome P450 here, SIP over here. Yeah, okay. So I don't know whether we didn't have the structure of P450 in the earlier slides, but over here this is actually the proximal view. So why proximal view? The classical understanding says that the substrate has to come inside through the distal pocket, okay, and then it will go inside. There was an image that was shown by Sir previously, I think. This is the one. This is the one. This is not the actual thing, but then just a representation where you can see the drug. Ideally, according to the prevailing hypothesis, should come through this way and sit inside this pocket for the reaction to happen, right? And yeah, this. So this was type 1 and type 2 binding that Sir was saying. Both of these requires that the substrate has to come inside, okay? Whether it be the type 1 binding, where actually the substrate is not interacting directly with the heme, or whether it is the type 2 binding where there is a direct interaction between the ligand and the heme center, right? Now, we are talking with respect to DROS, right? Murburn drives on dissolved reactive oxygen species. So we hypothesize that it's not only that this particular distal pocket has to be there and the binding has to be there so that the reaction can happen. No. We are talking about the diffusion of the radical species that can happen. And then for that, we studied the whole structure very systematically. And this is a list of the important cytochrome P450. There are so many. Okay, there are so many other than this, but these are some of the prominent ones. Now, how many are there? 4 by 4, 16 prominent ones. Okay, P450 is not SIP, but just for the reference, we have put it here. Other than that, you have 15 SIPs, 15 important SIPs, all of them. What do you see here? What is common? This is the proximal site, not the distal site. Okay, I just told you distal site is, according to the prevailing theory, the substrate should come inside from that side. What do you observe here? This green thing is actually the proximal cystine. And what is that proximal cystine? Proximal cystine is where the heme is sitting. Okay, the Fe, the porphyrin ring, is sitting on that, the cystine. Okay, so do you observe that in all of these? It's visible. It's evidently visible. And then you also can see heme in some of the cases, like you can see that in 2E1 and 2S6. These are some of the isoforms. So what does it say? It indicates, what does it indicate? It indicates that the radical which can be generated here can diffuse very comfortably through these places also. Okay. So we saw that common with all the SIPs. Fine. So this slide shows us that. SIPs have solvent accessible and dross reducible proximal thiolate. So that's what is here. Now, coming to the bigger question that was sitting with the bevy of xenobiotics, especially the drugs. Okay. What do you see here? So many drugs. What are the sizes? Different sizes. Of course, you have so many different P450s also. But then again, if you look at the structure inside, you will find that more or less most of them are having a similar kind of topography inside. Some of them have a bigger channel at the center, not throughout. Okay. So if you imagine, if you take this drug, trabectedin, or if you have ritonavir, these are humongous in sizes. Okay. Okay, fine. Ibuprofen is small, relatively much smaller. But if you talk about the molecule like trabectedin, such a huge molecule, how does it go inside? What have you studied earlier? In enzyme chemistry, what happens? Yeah, it has to open up, right? Side has to open up. So what will happen if trabectedin has to go inside? So the pocket itself is not that big. One thing. Other thing, even if it is that big, the enzyme has to open up like this. Totally open up and then allow the trabectedin to come inside and sit for the reaction to happen. Is that even possible? Thermodynamically feasible? Not feasible, right?
Now, the another aspect is also there. If that's wonderful. If that happens, yet another aspect. Then why is it that only certain molecules like this get metabolized? These are all substrates for this 2C9 only. All right, they are all substrates for 2C9. Now, if that opens up like that, then in that case, why are the other drug molecules not metabolized? That is even if it opens up. Yeah, and another thing. Sorry, I'm interrupting. One more second. Now, when I went to the Gordon Research Conference in US, it's a very prestigious conference on drug metabolism. I want to put his name on record. Gabriele Cruciani he is from Italy. He has got also a company in UK. He's a very big professor. So he was an invited speaker, and he was giving about how they open up like this. And he had some molecular simulation modeling, all that these people do. And I just stood up and I asked, "Excuse me, do you think this is feasible?" Exactly what he said. I want to document that very clearly what he did. He just heard the question like this, looked down, looked up, took some time. The heart spoke. You know what? I don't think so. I'm doing it because all of these people expect me to. That's what he said. And the remaining all days he was with me. I was with him or he was with me. He is a rich guy, you know. So we spent, and he said, "I like the way you do science." Yeah. So
this slide just to show you the different kinds of topologies of the substrates and the reasons that we have just discussed about. So yes, please.
No, it is possible.
It is possible. No, we are not saying it is not possible. Now, if you talk about trabectedin, a molecule like that, how can it go inside?
That's why I said. That's why he said something that is not.
Yes, there is, but the whole protein cannot open up. If the whole protein is opening up, then you need a chaperone every time the reaction has to happen.
No need. Actually,
No. Wonderful. That's why I intervened there, Professor Burra. The idea is that the more important question is, if it opens up, then why does it open up only to this molecules?
See, there are unknown things. That is one.
Wonderful. So that's what we are.
We cannot thought process to convince our hypothesis.
Hey, that's wonderful. No doubts about it.
So I have shown also in my PNS paper that same sequence, same structure, same amino acid sequence has 24 angstroms of deviation. So you cannot say that the domain motions are not possible.
No, never was it said that. Never said that. Never.
They are not even domain motions. They are active like completely changing distance.
No.
How are you?
Absolutely, nobody negates that, Prasad. Nobody negates that. The point is that the extent of opening up and the specific opening up that violates the concept of Occam's razor. That's all. It cannot, see, what we did is we tried to map these kinds of molecules with respect to the amino acids that guard the opening channel.
The very concept that I was telling yesterday that similar lacuna in understanding. Okay, similar lacuna in understanding of structural biology. Okay.
Yes, please.
Similar lacuna. Structural biology is also very much skewed in its current state of affairs. Okay, so similarly, I have proposed some new theories and engineering principles also into that. So based on that, I can explain many things like you are explaining from murburn concept. Okay, so the very basis is all the structures which are being explained functionally are based on XYZ coordinates, not the functional aspect. Nobody went into the aspect of first when we see head, oh, head does this function. Now nobody is talking like that. If head is on the top, it is head. So analogy I am trying to say. So we cannot say that how will you explain moonlighting proteins then? How will you explain promiscuity?
Look, that's what I'm saying. The promiscuity of redox enzymes is what we are approaching here. And here, what Abhinav said very clearly is not that it cannot move. We know, all of us know that there are so many proteins that move things. Lipid lipase, it has a flap. It opens up to accommodate the substrate. All these movements are known.
You are talking about retinol, right? And that retinol HIV protease, which is the enzyme there, that itself moves 7 angstroms down to push and break the substrate there.
Yes.
So 7 angstroms it is moving, and we have reasoned out what actually is what is the mechanics that is happening for that movement.
Now, let us come to the context here.
Yes.
The context is here. See, the reason that you are having a problem with this is you have published some papers that says that it's moving. We are not questioning that moving at all. The point that is being contested here is why only certain molecule will induce that movement. Our data is analyzing what are the amino acids that guard these things, and if there is something like that, there is a logic, we would have to be correlating it. We cannot find how one small molecule, one large molecule is opening up the same active site. There is no logic for that.
No. Why can't there be no logic there?
Wait. Here, we have an alternate explanation that is very beautifully, when we dock, the molecule docks at the surface, all these molecules dock at the surface, that's what is coming now. And this channel, it is a channel for the raws to come out. It is not for those small channel that is there.
Yeah.
It is.
Similar thing has
You go to the CYP3A4, it will be evident for it.
No, similar thing was explained by proton wires in serine proteases. So something like that. The proton wire is very similar to the...
No, proton wiring, all that.
No, it is also chemistry, right?
Yeah. See,
and
here...
the way the proton is moving towards the active site, okay, and breaking the peptide. Similar thing you are also telling in terms of the oxygen species. In a very broad way, I can tell. It has its specificities and all those things. I cannot go into that. I have no knowledge about that. But this is similar. Platform is similar, almost similar.
No, but here you
structurally, what exactly is happening, which is what is the driving force to open up, we still don't know, and we are ignoring that aspect of.
No, see, whether it is opening up or not there is some evidence for opening up in certain cases. Now, to imagine that these xenobiotics, certain xenobiotics, it opens up, certain xenobiotics, it doesn't open up, and these xenobiotics are of different molecular topology, sizes. This is one hypothesis that for all these there is some random logic. Now, for one simple thing is you compare the two hypotheses. What we are presenting is comparison of two hypotheses. One is an active site mediated hydroxylation. One is a topologically bound substrate mediated ROS mediated hydroxylation. What we are trying to present is the defense of a thesis wherein the Murburn model explains things much better in terms of Occam's razor. And what I quoted is people who have done simulations saying that it looks not feasible because it doesn't agree with the data. But whatever data we have with the dockings we have done, the simplest theory is what we are saying. We have not refuted the motility.
Let me update the current status.
Go ahead.
There is a huge technical issues to understand the dynamics of a protein. I can't identify a particular water experimentally. I am modeling the water there, right? So there are technical issues because of which we are given an opportunity to hypothesize. Okay, and similarly domain motions also I cannot have, except for NMR which has its own amino acid size limitations. I cannot go for understanding the dynamics. Okay, so because of these things we can hypothesize based on the existing knowledge.
Yes, the point is there is no need to go for all those simulations when I have a very simple theory that can explain with the given structure. That's the point. Why don't you see that? It doesn't need to open up. It's binding somewhere, the ROS comes out and reacts. Very simple. It doesn't need to go complex. This is a complex theory, what you're saying. We have a very simple theory.
No, complex is from human understanding. But if you are sitting with a protein, it is very easy for it to open up.
Wait. Listen to this. So, now this is what a scientific interaction looks like, and this is what is avoided in conferences. This conference is not like that. This symposium is not like that. What he and I are doing, this is what people should be doing. Understand? Don't run away from this. Don't ever run away. This is what this passionate, he is standing up for his works, his pursuit, and I am also doing that. This is what we are supposed to do. And this has gone away from scientific symposiums. I'm so sorry to say that. Yes, Prasad. So the point that we are saying, very simple. There are two hypotheses. One, all these proteins, there is no evolutionary mandate for a xenobiotic to bind and open up. There is no evolutionary mandate. If there is a lipase enzyme, the lipase has to have an evolutionary mandate. So the molecule comes, binds at the place where it is supposed to move, and it moves. Here there are diverse, show the topography of the molecules. And then here you see the two hypotheses. Here you see the two hypotheses. One hypothesis, the polar hydrophobic active site is for the raw stabilization. There is no hydrophilic material there. So the radical gets stabilized. It's like bobbing and stabilized there. Alright, there is no water there. So that it can, there is no extra proton or all that. So superoxide gets stabilized there and can give hydroxyl radical and all that later. Now that species, when it comes out, there are randomly bound molecules on its surface and they get reacted. And if these have, now we can explain with the new theory how these molecules can get, suppose 3A4 reacts with one drug, 2C9 doesn't react with that drug. If you study the active site process only, and if everything is supposed to go and occur there, why should 3A4 open up? Why should 2C9 not open up? 2C9 also opens up for all these bigger one. 3A4 also opens up that one. So why doesn't it? This here very clearly, when you dock also, it supports our data. So can we just move on? Can we continue with that later? Is that okay? Because the tea is already there. We have to finish this session. We will continue this. I like this. This is what we are supposed to do. Yeah. Yeah. Thank you.
Yeah. So getting back to the hypothesis, that's what we were talking about. We have shown that we have this proximal end where it's diffusible. Now this is just the explanation of the same thing. You can see you can have this is the proximal end. where you can have the electrons or the dross that can interact here, and then you can also have oxygen or dross that can go through the other channel from the distal side. So you can also have substrate that can go to the active side. Like we were saying, we are not negating it, but there are so many other substrates which cannot, which we have shown very clearly over here. You can see these are all sitting on the surface, right? This cannot go, and it's very improbable for this huge molecule to go inside such pocket. And what do we show here? There are channels. Like if this is the heme site, you have channels coming out from there, which can go to different regions, and you can have a major opening over here. So even if you see this major opening, you can see they are narrowing down at places. You can see this narrowing down here. This is just a representation for one kind of SIP. Varies from SIP to SIP. Okay.
So another thing, that other one is that here there were multiple researchers have published that it is iron oxo species, iron peroxo species, compound one, this and that. The new theory that we have proposed, it's a stochastic theory. It can accommodate all these observations. The Ross that can come in can get stabilized in multiple rows, multiple types. So it is not, we don't have a specific substrate mandate. It is a general burning mechanism. So this, we have 100 about cytochrome P50s in the liver, isozymes. Lactate dehydrogenase, how many? 2 monomers, right? Hemoglobin, 2 or 3 monomers. Here we have, this is monomeric protein, how many? Hundreds. So what is the different approach? What is the evolutionary approach? Make proteins with different topology. All right? Get the molecules to come and then generally burn them. And otherwise we are allowed to buy. Of course, that is the other theory, that it can open up and take in. That is lesser probable compared to this, because here it is agreeing with our docking, it is agreeing with our evolutionary mandate, and Occam's razor, complex, simple. This requires molecular intelligence, that okay, now something has come with a particular moiety, I have to open up. This doesn't require anything like that. All right. So that's why we'll get back. We'll have that discussion. Move it, because yeah. You want me to? Yeah. So quickly I'll go over the next slides. I was fired for this work. All right. Before I went to the lab, cytochrome P50 and reductase, there is an N-terminal part of it, and they were supposed to get together and enable them to do that protein-protein kissing and electron giving. All right. So first thing what I did is I made those 2 proteins without the N-term. They were quite happily. All right. So the hypothesis was that they enable anchoring and all that. So that's what I've shown. I've taken different preps, and I can get, even with the protein that lagged the N-terms, I could get the activity to approach the control values with the N-terms. So what it does is, I showed that the N-term has lysine and arginine and rich regions which can modulate cross. They are cationic, positively charged. So superoxide is negative at the interface. That's what is important. This simple idea is applied in so many things. Proteins have very simple repertoires of amino acids. So they've evolved to use those things. So here is the curious case of Benz-Bromrode. Have you heard of Benjamin Button? The Curious Case of Benjamin Button. Have you heard of that movie? Okay. So that is, that of course you have heard. It's our paper. कौन बोले पीछे से बोला क्या? Oh, okay. So here, when you study the Ki, somehow the values of Ki, which are supposed to be constants, they go much lower when you get at much lower enzyme, the substrate or inhibitor concentrations. So this violates the classical um, uh, understanding of dose response. Also, this benzbromone, Jeff Jones was working on this. He had multiple papers, and he said that it was a typical active site inhibition molecule. I showed that benzbromone also inhibited CYP3A4, CYP2D6, and CYP2E1. Very different isomer. So it was an interfacial radical scavenger. So vitamins on CYP2C9 activity. So anytime I had a lipophilic, vitamin E is lipophilic, vitamin C is hydrophilic. So I derivatized that to hydrophilic, both them. When whether it was vitamin C or vitamin E, when it was hydrophobic, the inhibition was profound. So basically, arguments in perspective is substrate binding to CYP. The diverse substrate diversity can be explained by the new model, intramolecular kinetic isotope effects. This is very important mechanistically. I'm running for time saving now because your snacks have come for the last half an hour. It'll go very, what do you call? Soggy? Yeah. Rancid is not right. That kind of rancidity is produced by liberated fatty acids. Yes. Acidity, no, that is not. So it will become soft. So intramolecular kinetic effects. So what is intramolecular kinetic effects? When you attach heavy atoms to some molecules and give it at different places, then you find the rates of reactions. Now what happens is, when you see H and D, what is D? H and D. What do I said? Kinetic isotope effects. What is H and D here? In kinetic isotope effects, what would H and D stand for? So this I am undoing myself, because if I am going to put it on this, my audience don't even know what H and D I will be showing. So that's not good. Kinetic isotope effects. H stands for hydrogen. D stands for deuterium. Who said that? Okay, you should say it, not when others are saying. All right, can you catch? Catch. Good. What's your name? Yamini. Good. So, what is the difference between H and D? Yes, one atomic mass unit is the difference. But in terms of kinetics, you can clearly understand the mechanism. So when they did kinetic isotope effects, it was showing that the molecule somehow was tumbling around. So the classical theory required it to be bound, and it was tumbling. So what is it? Bound or tumbling? Bound or tumbling. So they invented a concept called committed to catalysis, wherein it can be bound yet be tumbling in the air. What does it mean? You can have the cake and eat it too. Huh? So, do you understand what the consensus driven thing says? Secondary oxidations. They say once the substrate gets hydroxylated, the substrate goes out. But then how can something that is already hydroxylated get further hydroxylated? Something like that. So mobility, electron, see, there is only one reductase at very low concentration, where there are hundreds of different CYPs of different topology. You saw the different topologies. How can you explain that? Protein-protein complexation, long-term substrate binding, cannot explain. So you have to, what Prasad said was very right, that there can be probability, but you put all the n number of things around. It requires many things to be right. And here promiscuity of CPR, one CPR at very low concentration, CYPs at very high concentration. This is explained by the new theory, because reductase, you produce low amount of ROS, and those low amount of ROS are kept in check by high amount of CPR. They don't need to come one-to-one complexation. So compound 1 doesn't form water. So fundamentally, a sequentially ordered multimolecular scheme. So most of the xenobiotics do not induce their CYPs isozymes which convert them. So there is no evolutionary connection. Protein structures, proteins distribution, mutation studies. In the same conference, in Gordon Research Conference, Paul Hollenberg, he had done mutations on the surface. and he saw that some drugs were, their metabolic specificities were changed. So I asked him, Paul, don't you think that this this surface residues, my theory requires, the murburn theory requires, the molecule can bind at the surface and then that's it. That's enough. So when he mutated those residues, the activities was lost. So I said, don't you think this is explained by the murburn model? He said, yes. And that's it. He was honest, but it didn't go any further than that. You understand? So this is very important in drug-drug interactions. The same drug can activate, inhibit same other drug metabolic oxidation. So how can we explain that? This ROS comes out, interacts with this drug, can create a radical out of this drug that can interact, cross-react, all that. So that's how you can explain this. So I think I will stop it here because there are biological electron transfer and all that to be done. But I want you to go ahead and have your snacks and come back as soon as possible. All right? I will not dictate time, but please, thank you so much. But this is the kind of interaction. But there is a problem. You know, time also is something, multiple topics. So I will continue with. Please go ahead. Come back. How much time do you need? What is the time now? 11:06. What time will be assembled? 11:20 is good. 11:20. 11:20. 11:20 we start. Did you understand my argument?
No, I am thinking how to complement whatever you are saying. That has to be. This protein dynamics is very difficult to experimentally prove, and I don't have a technology where I can identify water role. That means I cannot identify even a simple movement of say acetyl group or something like that. So at that time I am proposing, I can go on telling many other theories to accept just, but experimentally, if your ROS is there, okay, it has to bring about some small changes at the electron density. Nowadays we talk about all these atoms as here, but actually electron cloud is there.
It's cloud.
So that cloud has all the potential to do whatever it wants. So it is not impossible for the protein, if it thinks, if its will is that I have to open, if I am destined to, I am a protein, I am destined to open up, and it is evolutionarily I am compelled to open up, then I will open up.
But the point here is there is no evolutionary mandate. And there is no specificity or selectivity mandate that we can probe or establish. But the other logic is, you know, what really happens is, Late. Seven. 13 into 7? No, அது விட அதிகம் இருக்கும். Oh, is 7 into 13 எவ்வளோ? 70 ah? Okay. 15 into 7 and எவ்வளோ?
இதுல வந்து 59 தான் சைன் பண்ணிருக்காங்க. அது போக எக்ஸ்ட்ரா வந்து ஒரு 2 4 5 பேர் தான் சைன் பண்ணாங்க.
Okay, 70. Yeah. My dear attendees, please, may I have your attention, please? We have Professor Yahya with us. Where is sir? Yeah. Sir, yeah, okay.
Okay, sir, I will ask Partha to upload in my drive. This is
Some people have again gone or what? இன்னும் போயிட்டாங்க. यही प्रॉब्लम होता है, लोगों ने जिंदगी भर काम कर लिया, क्या करें? But then he understood.
अच्छा हुआ
हैं?
रेडिकल की तरफ बढ़ जाता है.
वही ना प्रॉब्लम, वही ना, बंदा पूरा वही मॉडलिंग कर रहा है, इधर से हिला रहा है, उधर से हिला रहा है, हाथ हिला रहा है, पैर हिला रहा है, तो तुम कह रहे हो असंभव. हाँ सर. Yes sir. Professor Yahya, chemistry department, he has come. It was very hectic for him, yet he has come. This is yeah. So he would like to address you people for a few minutes. Thank you. The floor is his.
Thank you.
Yes, Mike. I'll give you the mic. Hello.
Thank you, Professor Murali, Manoj, for inviting me to this, as well as the Dean of your college. So he's the guest. Yeah. Anyway, it's my first visit to Amrita University at Timada campus. In fact, I want to tell my students over here, sitting over here, that take every chance as an opportunity to grow. Professor Murali Manoj was mentioning this about the opportunities to grow. So take every chance as an opportunity to grow. Unfortunately, we have been, during all our academic life, not only many of others who are sitting over here, my senior colleagues, my junior colleagues, our academic curriculum was structured between the walls, and we were not allowed to think out of the box. So this event, I felt like a chance that help you to think out of the box. In fact, 2 years ago, when we invited Professor Murali Manoj to our university to talk about murburn concept, some of the things that were some concepts were in my mind was matching exactly with his approach, not exactly the theory, but his approach. So I just want to share something that I noticed since yesterday evening. I'm sitting over here and listening to the talk. What you understand during this talk, I guess you understood life is primarily centered on chemistry, chemical reactions. Life is centered on chemical reactions. So when you talk about chemical reaction, the first principle in physical chemistry comes to explain the chemical reaction is chemical kinetics. Am I right? Chemical kinetics. So when the term chemical kinetics comes into your mind, what is the first aspect that deals with the energy changes in chemical kinetics? What is the energy changes? Delta G is there, yeah. Above that, whether a process, whether a reaction can take place or not. Which type of energy? Activation energy. Activation energy. So that is the concept that is formulated by the big person with big mustache. You know that. Who is that? Arrhenius. In 1903, he received the Nobel Prize, the second Nobel Prize in chemistry. So when you think about the concept of activation energy, how actually these all, not only the chemical kinetic theories in chemical kinetics, but also the thermodynamic concept. The next thing is the thermodynamics. Without kinetics and thermodynamics, you cannot explain the life process. So all those theories were formulated by considering the target system considered during the evolution of those theories were gaseous phase or dilute system. All those theories were developed from very dilute system or gaseous phase system. Now the same theory and the same equation we are applying to explain the condensed gel-like polymeric system in life. Will it be fruitful? Will it be fruitful? I don't think. Will it be fruitful? It's all, I don't have a big proof, but I think that the theoretical concept that developed based on the dilute and gaseous phase system, when you apply those exact theories to explain the condensed system in life, this is a gel-like system, polymeric macromolecules. How do you apply the same concept and theories to explain the life process? My opinion is the scientific world has to rethink.
It happened after the Nobel Prize laureate Archibald Hill in 1930. He did this. There was a contest between physical chemists and physiologists that membrane transport occurs by this ion differentiation at membrane level. The physical chemists said that it is by bulk phase phenomena, and they argued for colloidal chemistry, coacervate chemistry. The speaker yesterday, all they were from that group. They have been fighting from very long time. They have been fighting the biologists because it's easily visible even with the naked eye that cell is not a solution, but they still it is solution chemistry.
Then I invite the young brain to think about this concept. What actually leading to when you approach this thing? Our work is not directly related to the biological process, but we are working on macromolecules, electrically conducting polymers. We try to explain the chemistry of the material. Unfortunately, the scientific world has been dominated by physicists throughout the history. Like one example I'll tell you, those people who try to explain the brain memory are not chemists or biologists. They explain how the memory is being stored in our brain. There may be, but whatever the memoristic devices or those articles they are publishing, they were all based on dry state material. Using the dry state material, they are trying to explain the brain memory. How is it possible? If Murali, Manoj or myself has written a paper on brain memory and sent to a journal, it will be go to a physicist. They are deeply rooted in magnetic memory system, history system. But brain is made up of what? How can you avoid the concept of ions in brain function? In memoristic device, in any article you see, you cannot find the role of ions. They only talk about electrons, memoristic, the magnetic memory. So this is another one example. But actually, what we concluded, our research group, we have arrived in one conclusion that is not course, it is not hypothesized or discovered by myself. My collaborator, Professor Toribio Fernandez Otero, he is from Spain. So he come up with a concept that is called structural chemical kinetics. You don't find this word in any textbook. This is the foundation to explore how the biochemical reaction takes place. The basic concept is based on the conformational relaxation during the reaction. It's a macromolecule. So during the reaction, there is a conformational relaxation of the big polymeric chain. So you need certain amount of energy for that. That energy you cannot find in any thermodynamic equation. That energy, part of the energy is missing in delta G that you are applying in any theoretical equation. So this is called, this concept leads to another unexplored area that is related to equilibrium. So we know Le Chatelier's principle. Any system we say that it is approaching towards equilibrium, but most of the life functions they take outside the equilibrium. Am I right?
Yes. Well, if you see biochemistry textbooks, all the reactions are fundamentally occurring somewhere around delta G plus 0 is plus 10, 20 kilojoules per mole.
Deviation from equilibrium.
Yeah.
So then what is then how do you approach the Le Chatelier's principle into this life function? So the origin of the energy, biochemical reaction, always you find current and potential. There is a role of current and potential in biochemical reaction. So how this potential is related to the electrical energy changes during the that is the origin of the energy, conformational relaxation. So this, when you properly understand the structural electrochemistry, some of the questions, that was the one question he was mentioning, Professor Prasad was mentioning that nobody knows the origin, so I think that this conformational aspect, conformational structural relaxation, that we call every moment this polymeric molecule acts as a molecular motor. So this molecular motor functions due to the energy received or the potential received from the brain. So there is a current changes. Those things are not a subject of study by any research group. They approach the biochemical reaction keeping away the role of the potential arriving from the brain. So there are so many avenues. I think this is a very good chance for you to grow. This will help you to think out of the box. Don't keep your brain within the boundaries of the box. I wish all the best for the initiatives taken by Professor Murali, Manoj, as well as this university in this field. That will help. I expect that out of these around 80 students sitting in front of me, maybe 5 to 10 students can come up with marvelous ideas. Maybe after 40, 50 years, they will be in this classroom. I expect some of you will be, after 40 years, you will be well-renowned scientists in this area. All the best. All the best, sir.
Thank you. Thanks. So, thank you, Professor Yahya. Can we continue? Actually, what he said is very, 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 very true. We try to apply theories, prior available formats into the new problems or whatever we get. I'd start off with a class stinks. A class stinks. And they find the reason. Is it on? It is. In a small class, there's a girl with only one pair of socks. Unfortunate, economically challenged. So now they get an inspection. The education inspector is going to come to the school. So they all contribute. The class students all get together, contribute, and they buy a new pair of socks for that day. And the day comes, she wears the new socks. But the class still stinks. When the inspector goes off, the teacher gets mad. He says, what the hell, why was it stinking today? The girl comes up with the socks in her pocket and says, what should I do with this, madam? Do you understand? The Newton's first law, what is it? Things continue to be in state of rest or of uniform motion. One profound statement. It is very relevant. It has got its good things in our society. It has got its bad outcomes also. So, point is that we have certain ways of doing things. We have certain views of seeing things. And once certain tracks get laid for us, certain boxes, like what sir was saying, we choose to stick because it's comfortable. Comfort zone. Track, laid, move. So with that, let's go on. How can I get it to light up? Where is the... Okay. So, the drug-drug interaction part was where I was in. So, the same drug can activate, inhibit, all that. If it is binding based, difficult. If you invoke the diffusible radical reaction, then yes. Now, so biological electron transfers, when we talk about, what were we talking about earlier? Redox, electron transfer from reductase and things like that. So, here we did some further experiments in our lab. We see now, here we are probing the reaction of reductase giving electrons. So, this reductase gives the electrons not just to P450, to cytochrome C, to cytochrome B5, to about almost 100 different proteins it can give. So, how can we explain this with molecules coming together and trans-protein routes of molecular, between molecules, electrons going like a circuit? Rather than that, one protein, another protein, superoxide going. Oxygen takes electron, becomes superoxide. It interacts with ions. It can give electron, take electron. Very, very simple chemistry. Nothing can stop it. That's what. So that's what the data here is. Cytochrome C getting reduced from cytochrome P50 reductase, the electron source being NADPH. You can see cyanide inhibits that reaction. You can see copper messes with that reaction. Positively, negatively charged ions both mess with it. It's a very simple chemistry. Why? Because these interactive diffusible species that can affect it. And now you see what happens when you, earlier we probed that hydrophobic lipid phase reaction. Now we probe the same reaction where cytochrome C is in the which phase? Aqueous phase, or cytochrome C is a soluble protein. Cytochrome B5 is a lipid protein. Now here you see the effect is quite different. You get you see hormetic effects. The hydrophobic ones don't give that much. Hydrophilic ones give high effect. So where the reaction occurs, that's also important. Which molecule is getting the electron? That is also important. So understanding. So that's what this shows. I will not go into that. Then I added hydrophobic protein. So basically it's just a random collision based interactive equilibrium. I'm not going into these details. I've already. So even this reaction we separated with the dialysis membrane. We are getting electron transfer. So if it is, the theory is that proteins come like that Michelangelo's chapel, protein contact and electron transfer, if that was the thing, How can you explain these outcomes? You cannot. So yeah, I'll not. This is a very profound database wherein you see after some time the peroxide depletion stops, but when you add a substrate, ABTS, boom, the electron transfer continues. This clearly argues for outside active site interactive redox equilibriums affecting here. Why should the enzyme open up when there is something like that? Why not now? Why should the peroxide get depleted here like that? So cytochrome C, if you see, if it is by a topology driven complexation, you see the topology changes. Cytochrome B5 topology changes. How can you explain this? Topology is changing. So only the intermediacy of diffusible species can enable us to understand how diverse molecules, topography with different surface amino acids and all such. Why should this, if they are giving in a specific circuitry, why should this be solvent accessible, making it so chaotic? If it is, wiring is there within the protein, then why should it be like that? So the murburn ET is more probable. So here you see the data plotting with the classical theory, you don't get it beautiful like what you expect. See here, the molecules don't form the 100%. Sometimes the benzbromorone, which is an inhibitor, you see it is activating sometimes. All right, all that. This shows that it is not active site topography driven, and so here the molecules were, see, this was the molecule, right? This moiety is 2 halide or some substitution here, one thing here. So we took dichloro or dibromo, dihalo substituted molecules, and we probed the effect on the reactions. So when you do chemical mechanism probing, what you do is you basically alter structures here and there and try to understand what is the outcome. So here, when we did that, it doesn't make any sense. So here is, with respect to the classical theory, here is the theory that makes sense. That is, the molecule can get placed like this, and this can mess with the diffusible reactive species at the interface. That's what it is. So that completes the talk on cytochrome P450, biological electron transfers, and protein-protein complexations in a very simple 2 or 3 component system. What were the components in cytochrome P450 system? Can somebody tell me? Prabhakar, konjam idu pandringla, and the second talk vechidringla. Thank you. Can we interact some chocolate, or I'll eat my own chocolates? What were the components of the cytochrome P450 system?
And cytochrome P450 reductase.
Reductase. 2 major proteins and 1 auxiliary protein. What is that auxiliary protein? No. Cytochrome B5. Yes. And then in some systems there is also iron sulfur protein in this P450 system. So basically you have heme system, you have flavin system, and you have iron sulfur system, all that. All right. So what was the electron donor in this system? Good. What was the electron donor in the chloroperoxidase system? Peroxide or the substrate. Both can be electron donors. When ABTS is used, when you are trying to see where it's going, it can coming from both. So what gives the electron, where it goes, all that depends upon the mixture. So what happened in the P450 system? Heme was primary activator and flavin was just NADPH processing. Now here what we showed is it is not necessarily. The roles that were attributed were different. The mechanism that were attributed were different. So what we learned from chloroperoxidase we applied in P450 system. Now what we learned in chloroperoxidase and P450 system we can apply in respiration, mitochondrial. Every living being that uses oxygen, you, me, your parents, tiger, plants, bacteria, all. The most fundamental process is what? Respiration. After that, we will do photosynthesis. Photosynthesis is a more fundamental process than respiration. Why? Because there we are dealing with conversion of energy to fix that energy into something organic molecule in terms of redox energy in NADPH or molecule like ATP. That's a product of, and what else is photosynthesis product? Now that, of course, yeah, that is, of course, I'm looking for one other very important molecule that photosynthesis. I said one is NADPH, one is ATP, one is of course starch, whatever, fixing in carb. Yeah, one very important product of, who said oxygen? Yeah, oxygen. Oh, you are a plant physiologist. No mark for you. She's a plant physiologist. So, yeah, good one. So, yeah. No. My standards are not that. She's a teacher, no? You're a teacher, right? Yes, she is a teacher, but she's a plant physiologist also. So, no chocolate for that. Yeah, but that is very correct. Oxygen, without oxygen, we will not survive. So, yeah. So basically, we are now going to, now even, was xenobiotic metabolism not important? How will, is everything that we take in, everything that we are exposed, is it, that goes, that is also important. In fact, xenobiotic metabolism was the primary way to get energy. Then after some time, more well established ways coming in. We are trying to understand life, mechanisms of life. Now welcome to the most important procedure in life, that is respiration. Respiration and thermogenesis. What is thermogenesis? Generation of heat. What is the body temperature? 37. What will happen if you are 20 degree? Death. Good. Now, so that is also important, no? So why is, in chemical perspective, why is 37 degree important? Why 20 is not? Chemical. No. Why is heat required for body? Chemical answer I want.
All the reactions require some amount of heat or energy.
Why?
Reactions. We have to break one bond. We need some energy to break that.
Yeah, bond. Okay, let's not go there. Bond. What? When reactions occur.
Maximum rate of reaction is highest.
No, yeah, but then why is rate dependent on temperature? What happens with?
Depending on the.
No, what happens when you have two molecules reacting? When temperature increases, what happens?
The motion of the molecules increases.
What happens because of that?
The probability of collision also.
Ah, that is nice. Collision theory. You remember my first slide, first talk? My world was collision theory, active site, transition state theory, all that, right? So temperature, heat energy is important. Now our body needs to produce chemical. We need to get powered. Anything does useful work needs to be powered. Now we are fundamental powering mechanism of life forms. All right, wonderful. So this is an old slide. I am not using my new slides. I deliberately went for my old slide. Now, why am I using this old slide? To show you that I had said certain things in 2018 or 17. This is, when was it? Who prepared this, Who prepared this slide? You did. Vivian David Jacob, he prepared this. Do you like it? Do you like what you see? What is it? How many Mays in how many years have come and gone after this? 7. Yeah? 7 Mays, right? Yes, 7 years. 7 Mays have gone after that. 19th of May 2017, Molecular Biophysics Unit of IASC, the cream of our country. All right. So, Carolyn Wells, that, okay, so did you understand this poster? You understood? What about you people out there? Is it sensible? It is way to get your attention. When I critique a Nobel, that means the idea is important. So it was something in 2017. Now, all right. If you are with me, you would have gotten certain things. We used chloroperoxidase system to understand P450 system. And the P450 system is a complex system of flavin enzymes, heme enzymes, and some soluble things in a lipid membrane, right? Now, what is the similarity between, where do you think respiration occurs? At a mitochondrion, right? The cellular organelle. What is it? It is a lipid-laden, convoluted structure, right? Now, we have already said that lipids are oxygen-centered reaction places because radicals can be more stable there. Hydrophobic pocket of SIPs, the lipid membrane per se, all that. So, both in SIP system and in the respiratory mitochondrial system, you have low flavin, high heme, phospholipid interface, NADPH and O2. The reaction components are exactly same. Only thing is, in the cell, the endoplasmic reticulum, where xenobiotic metabolism occurs, that is not a closed structure. It's a open membrane. On that membrane, this side, that side, it can happen. Here, so prolonged, both prevailing mechanisms required prolonged substrate binding, that is oxygen binds to cytochrome oxidase, sits there, 4 electrons, 4 protons to make water. You know what making water is? Literally? No, you don't know? Making water is? No, making water means literally urinate. Alright. Alright. So, to imagine that oxygen is molecular urinating mechanism, it's a travesty, I would say. I'm deliberately saying this because people are not accepting my theories, so I would rather mock them because they call my works controversial when it is thermodynamically sound. So I'd rather mock those people. So I am putting it out there. So all required proton consumption, proton heme forms water, protein complexation, ROS or side products, deleterious, varying stoichiometry were reported. So many systems were similar. And experimental observation, in the SIP system, you monitor NADPH and its depletion was like this. Microsomes, you add NADPH, small rate, you add the substrate, boom. You remember Abhinav's data with simple enzyme, when you add substrate, even just now I showed, it goes down quickly, the thermodynamic pull, that was here. And when the depletion, the substrate is converted, you have the same NADPH depletion. In the mitochondrial system, you monitor oxygen consumption because it's a complex system, membrane laden. NADPH is 340, 350, around that nanometer. There are so many things interfere. So experimentally, you monitor oxygen evolution by oxygen electrode. That's what is done. You see the profile is exactly the same. So in mitochondria also, several SIPs are seen and non-specific phosphorylation of xenobiotics is seen, although uncommon. So there are lots of similarities. Did you understand what I am trying to point out here?
Can you repeat the statement?
Which one?
The one that you just...
See, what I am trying to show is there are a lot of similarities between the xenobiotic metabolism and oxidative phosphorylation that occurs in mitochondria. Oxidative phosphorylation. What is oxidative phosphorylation? Using oxygen, ADP gets phosphorylated to make, and that is the powering energy molecule which all of us use, right? So here, now you see what is the classical theory. You have seen the video, yes or not? Yesterday. What was the video? Lovely. All this goes nice, hunky-dory, all those nice things, molecules go, I will go from here to there like this. I will from here to there, I'll come like this. Do you think molecules are destined to go like that? No. That's the first thing. When you are a biologist trying to approach chemistry, they have their free wills. They do whatever they jolly please. We can only, even if I say 11:20, you have your snacks and come back. Will you come back? You have a control mechanism. You are not going to come back. That's what entropy or chaos is. Basically freedom is that. So, no, I didn't use the word entropy correctly there, but anyway. So basically, the theoretical depiction shows that there are a bunch of complexes. There are a bunch of movable molecules like CoQ and cytochrome C. Electrons are put into the system and it's like a downward slope wherein oxygen stays wedded to complex 4. Happy sits there like a good boy or, good girl waiting for the 4 brides of, protons and 4 Dahej of 4 electrons to coming just like that. I can't think of better things because I have to mock these people. They cannot continue. It is criminal. Scientists have only 2 things required: intellect and integrity. It is criminal. I put that first slide. 2017, 7 years, I'm talking about oxygen utilization. How many people died in COVID? If we knew how oxygen is utilized, how many people could have been saved? Right? Do you think it's a silly thing that I'm talking about? It is not. It is more important than the Solvay process, all that. Now you understand what I'm saying. Now you see. And oxidative phosphorylation has nothing to do with oxygen. How ATP is made? This proton that goes out then creates a gradient. And then the protons come in. Ghuma ke. Complex 5 goes round and round. And you have. So, oxidative phosphorylation is an oxidation process and a phosphorylation process. It is not really oxidative phosphorylation. There are 2 distinct steps. You see that. And what this is there is a negative relative charge inside because protons are pumped from inside to outside. That is the reason for the electrical activity. And this is positive, so it comes in. Alright, so this is, all these ideas have, even the Marcus theory was applied for electron transport and protein-protein complexation and all that. So, multiple. So this is it. Whatever you saw in the video, all that is here. This is complex one. NADPH gives 2 electrons. The CoQ is what? It's a isoprenoid long quinone. So I take electron from here. Now it is my heavenly bound duty to take this molecule, 2 electrons and 2 protons. I must take it to the cytochrome, your complex 3. I have to take it, please. I have to. And then there it goes. And then the other guy comes. Now the protein guy comes. Protein says, I have to take it from here to somewhere else. And then from there, all that, I want to make water. All that for that, okay? You guys, you understand, no? All that for such a wasteful purpose, okay? So, and in the meanwhile, this guy sits waiting for the protons to come. And these are the complex equations and the 2 things and overall. So, how much ATP it can make are determined by Nobel laureates. Earlier it was 4, then it was 3, and all like that. I'm basically kicking my own butt when I say like this, but I hope one of you people will take all these things seriously and go ahead with finding the truth here. So, the way it is depicted in textbooks, it is like this. The redox potential, as the electrons go down some gradient, there are 3 steps where ATP is made. Where is this electron going? How is it correlating to the 3 steps? You don't know. But theoretically, some idea is just kept there. And each one of these steps, it can be stopped by some inhibitor. But then ATP is made by what? Complex 5. Does this inhibitor affect that? No. I don't know. How it's connected? No. Some connection. So some theory has been given to us. Remember my Escher's image? Perpetual water. You can make things in images and you can give it. Whether you buy it or not, nahi, ye baat hajam nahi hui, I don't digest it. You have, as a scientist, it's your moral obligation to question, to doubt, to explore. So, working analogy is, now you have a potential energy harvesting in the dam hydroelectric power plant. So, I make a comparison with that. You can, the classical powering mechanism in life, you can see water is stored here at a higher level, and then water, when it comes in, it rotates a turbine, and that makes electricity. Mechanical is converted to electrical. Yeah, wonderful. This is the kind of thing that I imagine Peter Mitchell kind of, ruminated or ideated upon. And then there's one small problem. The small problem is the water that comes here has to go there again. The same water that has come here, because the protons are getting pumped again, and the protons are pumped out. So now you try to imagine something like this. You take money from your bank. You took out $20 or 20 rupees. Why should I say dollars? 20 rupees. 20 rupees from your account and from your checking account. Your bank is so nice. They put $60, 60 rupees in your checking account. Savings account से तुमने लिया, हम आपको 60 rupees checking account में डालेंगे. That is the wonderful theory that we have here. That is, when protons are pumped out, it develops an electrical as well as chemical difference, and so that is a big difference. And then that is simultaneously coming back also. So gradient है क्या? नहीं है, लेकिन है. It is there. So I know very well what I'm doing. I know I'm making faces. I am taunting big powers, but that is needed. While what they do is disregard truth, I'm just mocking. That I'm entitled. Now another analogy is the alternator in your car. See, the engine burns fuel, and then you know how alternator uses its the rotations to charge up the battery, right? Something like that. You know, the rotations can be proton pumps is the recycling logic, engine burning fuel and all that. So the only problem is, like Laurent said, how many complexes per mitochondrion? Any idea? 1, 10, yeah, there are 4, 5 complexes, that is right. But then how many numbers? Complex 1, complex 3, complex 4, complex 2, complex 5. How many of them are there? 10 to the power 5, something like that, per 1 mitochondrion. More, 10 to the power 6, it can get there. My own article says, I think, 10 to the power 6. So, 10 to the power 4 to 10 to the power 5. Okay, conservative proton requirement. You need at least 10 times more as per their theory at one instant. Okay. No, it requires 3 times higher as per Peyton Mitchell. How many protons in mitochondrion? You can calculate. What is the volume of a mitochondrion? 10 to the power minus 16 liters. So, what is the pH? Around 10 to the power minus 8 or 10 to the power minus 7. That H plus concentration. Multiplied by Avogadro's number, you will get the actual number of protons. So, it's 1 to 10. So, you have proteins this much, protons non-existent. How can it pump protons? This fundamental thing, Raja Nanga hai, what I told you, they gave Nobel without even checking this. Why? Why? After the Second World War, Anglo-Americans were powerful. Nobody could really question them. Actually, there were people who questioned them. The major person who questioned this was actually somebody from Australia. He died a couple of years back. If he was alive, this thing would be already in textbooks now. Because he was, but then all these people, they are still hand in glove. They accept. Political people, they don't have any animosity or things like that. They'll have no problem. So evolutionary mandates. The classical scheme is vitally deterministic and irreducibly complex. It requires 3 phases, multiple proteins, multiple molecules here and there. All should be controlled, coordinated. No. Losing end gametes. First, I shall pump protons out. Then I'll use some other machinery to make ATP. What kind of it is that? Is he an investment banker or something? What? What is happening here? Then I have very low efficiency. Why should ROS formation occur in mitochondria? Why is complex 5 when it is isolated? Why is it an ATPase? Why is it not an ATP synthase? Why? It's surely a mirage to say that, oh yeah, when you isolate it, it is a ATPase. But in the mitochondrion, somehow it gets emotionally charged and becomes ATP synthase. Then in that case, why do higher metabolic rate animals, all those animals that have higher metabolic rates, they die faster? So, why? They are very profound questions. What is oxygen's role? Oxygen's role is just sitting wedded to make water, like I said. And the point is, oxygen's coverage is easily around 10 to the power 3 angstrom per microsecond. It moves, tuck, tuck, tuck. It's very fast moving. It's a gaseous molecule in that domain. Now, biological ET, you will get the slides. I'll share the slides with you. You will have all the slides. Don't worry. So, biological ET is 10 to the more than 10 angstroms and in microseconds. Do you think whatever the electronic circuitry that these people have talked about till now, I have only used their data, the information that they have generated. I've clearly disclaimed that Tycho Brahe's data Kepler used. I didn't have much. What I'm saying is, if this is true, that oxygen can move this much in 10 to the power 3 angstroms in microsecond, and if the reactions occur in milliseconds at 10 angstrom scales, will not oxygen shunt all these supposed electron transfer reactions? Don't you think? Oxygen is highly electronegative. It wants the electron. I told you oxygen plus electron is minus 250 kilojoules to make superoxide. Why should a chemical give an electron? I will not give to oxygen. No, I will not give. इसकी मर्जी है क्या? It cannot. It is not within that. All right. So the thing that you see this, the molecules with that alcohol we just discussed in the last. They said that protons, when they put a molecule like this dinitrophenol, they could see the loss of redox equivalents, but no ATP synthesis. So Peter Mitchell gave a wonderful explanation. What? This proton that is there, that has been pumped out, it goes and takes and comes and gives it here. One side takes it, gives it here. And like all the other molecules, dinitrophenol also has been given a mandate. Protons cannot transfer themselves from here. What is the dimension of a proton? 0.00002 angstroms, around that. And what is the dimension of this guy? Several angstroms. What is the charge on a proton? +1. What is the charge on these things? Nitro groups. They have positive nitrogen, oxygen, negative, multiple charges. Huge molecules. Protons cannot go a small one, but multi-charged things can easily go up and down on a lipid. Do you buy that? Does it appeal to you in terms of molecular chemistry? No. But then some people said they had power, and there were people who bought it. So the distribution, if these, while the electrons are going through this and the protons are getting powered, why do I have lot of proteins hanging down from the membrane? Why are the structures like this? Why are the distributions so poor? That is, the flavoproteins is very low, like P450 reductase, very low, whereas heme proteins are very high. Why? If you imagine it's a serial transport, you will imagine 1, 1, 1, 1, so that the stoichiometry is maintained, facilitated, right? It is not. So when you do the basic calculation of the thermodynamics, at each step they cannot explain how if a process is facile, it needs to be having delta G negative, right? The calculations don't show that. Only complex 2 has a -32, all the other ones have, complex 2 and complex 4 have negative. Overall, it is not feasible. At other complexes, it is not feasible. All right. So here is something that I want to show you. I think we will come to this again. Here is the electron transport chain, multiple iron, sulfur, flavin, all that. This is complex 1. Electron transport occurs here, proton pump occurs here. No connection. Multiple electron transport, iron sulfur centers. Some are not in roots, and they are not having. What did we say? Electron transfer can occur from -300 to -200. So you would expect this is all nice hunky dory, -300, -250, -200, -150, like that, right? Something like that in a gradation. It's not like that either. So if you imagine that it has to be a proton pumping mechanism based on electron transport, so you would have redox centers here and the proton pumping like this. Why would you have a structure like this? such a huge pendulous with so many redox centers. Why? So then Q cycle is yet another Peter Mitchell's wonderful idea. Sorry, Peter, I like you. You are a nice guy, but as far as ideas are concerned, this is as bad as it gets. This here, this molecule, you see, you imagine like this. What does it require? It requires quinol to bind at one place. It requires quinone, the product, also to bind to another place. And then cytochrome C should bind yet another place. I'm doing all this. You know why? Why? To get your attention. Some of you would be going, so there's this guy doing something funny. Let's see. All right. So, but then this is also one thing has to carry. And so now you imagine I have an inclined plane and I'm rolling two balls from that. Two electrons have come from quinol. So it comes down. Then one thing goes somewhere there and goes down. The other one comes all the way up, back. One electron comes back to क्यों भाई आ गया आता है it just comes. That's a theory. Why? Because it explains. It explains. How does it explain? Because some people said and some people said. So now if there is a circuitry like that for electrons, would you imagine these heme centers to be so openly exposed to the solvent and membrane and all that? If there is a circuitry like that hardwired, you would not imagine that. And very interestingly, the KD is approximately 0.1 millimolar. That is the binding affinity as determined experimentally. But the Km is less than 1 micromolar. No, you just imagine this. Km is defined as Kd plus K2 by K1. So theoretically, is it possible? Km is in micromolar, Kd is in roughly somewhat lower than millimolar. Is it possible? So is it okay if I mock them? Yes. Yeah. So, and even then when they transfer even within the heme, in the anaerobic thing, when they try to check the electron transfer from one redox center to another, in this complex 4, cytochrome oxidase, the most important protein in the redox transport chain. In the anaerobic state, the electron transport was 1,000th of what is in the oxygen state, when oxygen is present. So oxygen is the one that transports electrons, is very clearly seen. So the structures of the components, all right, is here. You see these structures. This is coenzyme Q. This is such a long one. If coenzyme Q is the relayer of electrons, there are many different chain lengths. So this is coenzyme Q10. This is the most abundant one. If you wanted it to relay electrons, what would you imagine? It should be smaller, no? This tail should be smaller so that it can go quickly. So cytochrome C, once again the heme is almost exposed. So these are all. Now here, what is shown is, we show that here it also has ADP binding site. Abhinav, I told him, Abhinav, check if all these complexes have ADP binding sites. Voila, they have ADP binding sites. And it was also experimentally reported. And we checked their ADP binding sites. Beautiful. Do you know that this complex 3 in the classical model, ye jo sac jaise, this is hanging down, there's no rule. No rule. No role. Huge part has no role whatsoever. So this also has no role. This of course just takes electrons and gives it here as per them. then these things. So now our new theory is basically very simple. Now I'll come to those images. I think it is better if I show them what the murburn theory is. I would not go too much into details. Murburn theory says all these complexes 1 to 4 have redox centers that can activate oxygen and give ROS, and they also have ADP sites. So ROS can attack ADP and phosphate, and by the 1 electron route they can make ATP, and that porters and porins can take the ATP and water out because that's a positive pressure there inside, and it goes out. The uncoupling protein can mess with the ROS and lead to heat. So the murburn model has direct attack of oxygen-centered radical with this. There's direct, tangible, chemical, bimolecular, kinetically viable connectivity. Did you understand this part? I was given a criticism that when I talk about these fields, I do not talk about my model. I spend more time critiquing others' model. My model is only required if the other model is wrong. Yes or no?
Yes.
Why do they criticize me for criticizing them? Because they go out of business. Right? Don't listen to such political statements. Scientists, you don't care. You argue. You are a friend, don't care. You argue. You are a teacher, you don't care. You argue. As long as, but you might not get a job in the same department because I found it out. That will happen. You say, oh, sir, yes, sir. Then you will get in the same department. But then you will not go far in life with that. Once you compromise and get that, you are compromising with your spearheaded attitude. And those kind of skills that you hone over a period of time, it doesn't go. See, I'm a biologist. I don't know a thing. I don't even, I'm honestly confessing on video, audio, very clearly. All those people who work in this area, my information skill or conceptual level is 1,000th of them, I would say. Their awareness is too high. But I'm just bullheaded. I do not buy nonsense, whether you serve it this way, paint it with, BS, you paint it with nice golden colored spray paint and all that. No. It is BS, right? So that is it. So this is the murburn theory. So homeostasis is easy, thermogenesis is easy, and transmembrane potential is explained by what? What is transmembrane potential here resulting from? Can anybody say by now? Why is the negative charge resulting inside? Is it because of proton pumping? What is it because of? You have already attended first talk. He said that very clearly. It's a chocolate question. Loud and clear. Now she's seeing that. Say loud. ROS. What are ROS? Which ROS? Which type of ROS? Can you give me one example of ROS that can give a negative thing inside? Oh, lovely. What's your name? Christina. Yes. You are from Thrissur. Yes. Good girl. So that's a very fundamental thing. That is what gives you the electrical activity. Fundamental. It's not ion pumping. It is effective charge separation. How? I told you, giving you a chocolate doesn't take away my beauty. I am the beautiful person. You have to always have your eyes on me. Because from here comes the information, all that. You lose that fundamental, gone. Okay? Now good. So, these are the kinds of reactions that happen. See, all tongue. At complex 1, it can give, and so in this reaction, what is the role of cytochrome C and ubiquinone? Ubiquinone is an electron trapping mechanism randomly packed in the lipid. It is not something that takes electron from one place and goes. Oxygen is free to move around. Cytochrome C absorbs the electrons that comes in the aqueous phase. All these are just effective charge separating mechanism and ADP presenting mechanism. Is it similar to the xenobiotic metabolism principle? Exactly the same thing. Enhancing the probability, the protein merely enhances the probability of the radical to interact with the substrate. That's all. Otherwise, the radical will find another radical or a more potent electron donor and react with it. That's a probability gain. Very simple. So all these reactions, they are high kinetic viability radical reactions, bimolecular, where these people's reactions had one catching, another catching, another catching, another catching, 5, 6, and then still not happy. What is the probability of these things happening? Very low. Even if you consider bound, unbound state, 1 by 2, 1 by 2, 1 by 2, then 1 by 2, probability of another molecule binding. Multiply. If you have to have 10, how many steps? Dozens of steps in their scheme. So the probability of that thing happening is insignificant, improbable. Yeah. So as oxygen keeps accepting from electrons, becomes superoxide, basically peroxide ion or hydroperoxide to hydroxyl radical, hydroxide ion to water, these things happen. So it is not oxygen staying wedded at complex 4, but oxygen doing its magic as diffusible reactive species, that we are existing as living beings. If they are not allowed to do with their random movements, we will not have effective charge separation. So you see here, the overall thing is like that. Oxygen reacts at all states, and then you have this becomes proton deficient. So protons can be used from outside, and then the uncoupling protein can porons. This ATP can also get synthesized here, but it's by a different mechanism. I will come to that. So now the structural aspects that supported the theory was, I predicted, and Abhinav looked at it, and we got wonderful data that all the complexes bind ATP. When we search for it, the data in literature, wonderful. They already had reported that. So they didn't think that this could be the reason. So this was eukaryotic system. So when I went to IISc, one wonderful girl, she said, if your theory is true, you should see this in prokaryotic systems also. Wonderful. Prokaryotic system. All right, same like that. Multiple ADP binding sites on both this. And they have pores. And they have all these non-root redox centers, not in the right sequence. Deliberate mechanism for generating what? For generating what? No, reactive oxygen species, right? Then, and these reactive oxygen species, the ADP binding sites will enable a low affinity way of ADP to come transiently bind and get the attack, all that. So that is what these structures are for. So, but then here it can use the electrons from coenzyme Q to give it to oxygen. Here it uses peroxide. This is just a peroxidase, and that's it. Peroxidase, it will pull the equilibrium further down and sink the electrons into water. So because oxygen, peroxide, water, right? That's the way. Now structures. So here are all the 4 redox proteins. So erstwhile structure function, coenzyme Q comes out of the membrane to take electron here. That is what the role. What it should be, and the structure should have been like this if their theory was right. What is the new attribute? Absolutely agrees with this structure. So similarly for complex 2, complex 3, complex 4. All right. So yes. So this is a big farce. Paul Boyer was not a good scientist, Caltech professor. When Edward Slater, the person who opposed Peter Mitchell theory, he actually was saying that a phosphorylation ping pong mechanism enzyme intermediate existed. So they tried to hunt for that intermediate. They could never find it. When Paul Boyer, whose rotary ATP synthesis mechanism was in later, earlier when Ed Slater was big and influential, he said phosphohistidine and published it in Science. So all these people with big time labs, they can discuss அவனுக்கு ஒன்றும் தெரியாது இதை பத்தி எனக்கு தான் தெரியும் ரைட்டா இல்ல ஏதாச்சும் சொல்றேன்னா ஓப்பன்லி சொல்லிடு எஸ் அவன் கிட்ட கேட்ட அவன் என்ன சொல்லுவான் என் கிட்ட கேள்வி நல்ல கேள்வி எஸ் நல்ல கேள்வி I said I promise that those people who ask good questions will get chocolates. I'll come to that answer after some time. All right, good question. அவன் சொல்ல முடியாது கேன் சொல்லி அவனுக்கு என்ன ஏய் பேசாம மூடி கம்னுறான்னு சொல்லி அப்புறம் என்ன you understand no you get only நாளைக்கு afternoon after that I'm out of your life. All right, if people start accepting my ideas I won't even see you. yaar yaar all right, so no just kidding, but yeah I won't see you, okay? Because too much time you have to shake up a bit, laughter, some people will be sleeping. What did I miss? So this thing Paul Boyer said, clockwise protons come from here to there, ATP breaking, anti-clockwise or something, ATP synthesizing. Good, nice idea, only sorry feasible nahi hai, it's nice, not working. So the yield substrate level phosphorylation is even better. Why should evolution go for such a complex thing? Why should it go for all this? You know what is substrate level phosphorylation? Phosphorylated molecule gives another molecule, it's phosphate. So that is substrate level phosphorylation. So see, there are publishers, there are experimental observations which say 3 ATP per NADPH, NADH is observed. The Nobel laureates come and say, no, those are all wrong observations. 2.5 is the maximum we can give, okay? That is where, stomping authority over. In science, it is the authority of logic that counts. It is not the, what did I say now? It is not the logic of authority that counts, it is the authority of logic that counts. Did you understand the play of words? Logic of authority. Nobel laureate is the authority. Who is the authority actually? What is the authority? Logic. And that logic is within each of us. Alright, of course somebody will have crazy logic also, but then let us not go there. Alright, this very important, the mirage. If there is a pH gradient in the working system, as Peter Mitchell said, you should be able to observe it. There was no pH gradient. There was nothing observed. And complex 5's affinity is much higher for ATP. So in mitochondria, there is more ATP, 10 times more ATP than there is ADP. And it has more higher affinity for ATP. So what will it do? An enzyme is named after what? Its substrate, yes or no?
Substrate and its mechanism.
Yeah. So if I say an enzyme is using manga as substrate, with tenga as cofactor, and with katrika as product, what will be the enzyme's name? Enadhu sonna?
Mateka enzyme.
Matenga.
Matenga enzyme.
Matenga No chocolate. No, no. Authority. No. Here, authority is the logic. Now, matenga. No. Manga is the substrate. Tenga is the product. Katrika is the cofactor. What? Chutney. No, that is the process of making chutney, you can say. Good one. But innovative way of thinking, of course. It doesn't get the chocolate. We can all laugh. Good. Yes. Manga katrika enzyme. No. An enzyme acts on sucrose. What is it called? Basic, simple. Whatever is the substrate for which it has affinity, manganese. For this question, I was called, I asked this, VIT, I was called by the Pro-Chancellor. I had to sit there for 1.5 hours trying to explain. My management said, these students are all, some people had written complaints. Students are from different parts of the country. How do they know what is manga, what is tenga, what is katrika? I said, I taught them bloody hell. So, anyway. So, it's an ATPase. Damn it. And then, if the ATP is the substrate, you call it ATPase. So, it has got higher affinity for ATP. Damn it. So, and also TMP is continuing. Oh, okay. TMP is created, transmembrane potential is created, then it is dissipated. Okay. Ye kya hai? It is getting pumped out and coming back in at the same time. Where is the gradient? There is no gradient. It's just a marichika. Mirage has been created. It's just an eyewash. Exactly what? Do you see what is happening here? Water goes downhill, and comes down, and then drives the wheel, goes downhill. Is it possible? Is it possible? No, it is not possible. Right? So that is where murburn comes. All this is beautiful. And our Surjith now will show you something. He did the work at IIT Guwahati. It's his moment. I'll just tell this and then you go. Get your. If you go, you cannot. I cannot show this, right? Mutually exclusive events. So yeah. So here is something beautiful. This is Nichols' aging cell paper. He's an authority. Nichols, if you're listening, it's for you. Wonderful data. Here it is. When reactive oxygen species is high, observable, only then ATP synthesis is observable. Only then transmembrane potential is adequate. So when the vehicle runs, you get sound and smoke. The vehicle runs because of sound and smoke. Is that a good theory? The transmembrane potential is because there is effective charge separation. ATP gets synthesized because, and that here this is the equation in my theory. The ROS H2O2 is also a ROS. It is not ADP plus PI gives ATP plus water. It is also producing ROS. This is a charge mass balanced equation. See how kinetically and how energetically viable it is. Easily you can produce perfect. This cannot be wrong because it is starting with ab initio calculations based on delta G of formation of the molecules and the products. All right. You have the numbers here. You have the numbers here, and then you can calculate. But anyway, this number you can also. I have taken the proton to be present here. If you take 79 kilojoules for water dissociation, then you will get 3 or 4 only. But theoretically, it is very facile. So this is what the data is very clearly supporting the murburn theory. And also, you give mitochondria NADH, more ROS. You give mitochondria more oxygen, more ROS. So the mitochondrial system is geared for production of ROS. Increase in ROS elevates TMP, transmembrane protein. ATP production is not seen without ROS. And then complex 1 to 4 all are known to generate ROS and all are known to bind ADP. What more does murburn theory want? Everything is happily satisfied. And we now know, thanks to Abhinav's work, we know where it binds also. And it also agrees. Wonderful. And so this is the equation. In vitro, Surjith showed, he'll show how he did that. No, I was not the first guy. We were not the first guy. There was wonderful women scientists, Kathleen Mailer in 1990, and Mildred Cohn in 1950s, Galina Mironova. Galina Mironova is still kicking. She's in Russia. I wrote to her, asked for something. She wrote back. She showed peroxidative phosphorylation in 1970s. And Mildred Cohn showed using radioactive, heavy oxygen, she showed that when ATP gets formed, multiple O atoms with labeling get incorporated. Radical attack, tack, 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 tack. It's not tachkum, tachkum. Not like that. Tung, 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 tung. No, that's how I see. That's how I imagine. I'm a simple guy. I'm not a very sophisticated guy. I told you, these kind of things are for, science is for simple, straight thinking people. All right? So, that's clearly indicated radical reactions, which I don't know how Paul Boyer interpreted for his theory and how other people bought it. Anyway, Mailer showed you take mitochondria and just give xanthine and xanthine oxidase. They are known to produce superoxide. You get ATP. All right? Also chloroplasts. Now, Surjith will take over from here. His work is about how he did that work in IIT Guwahati. It was not his PhD area. It was completely different. What were you working on for your PhD, Surjith? Bioprocess. He did something for me and for himself, curiosity. We talked and he did it. So, yeah. Special effects. Ta-dan. He did something for me. Ta-dan, ta-dan. Yes.
Good afternoon, everyone. Am I audible? Yes. Okay. First, let me tell. Okay. First, let me tell how did I meet Murali sir. How things have happened. Like first, when I was doing my MTech in Industrial Biotech in Shastra University, I think so. We were in first semester. We have completed first. We were going to second semester. We were given elective courses to choose, like some food technology. Some things were there. So me and one of my friend have discussed. One guy is there. He has come from IIT Madras. He has postdoc experience from outside. If we study some course from him, he was offering animal biotechnology. It will be very good. I also had very much interest. I also want to study more about animal biotechnology, and also I am very much interested in bioprocess. Animal cells also I want to read. So we have planned. Okay, let us go to him. We will do something. We will learn something nicely. Like that we have thought. So we were of 22 people in the class. We have asked everyone. They have already inquired about him from other like juniors. They have told we don't want to ruin our CGPA. Okay, because though everybody, they have scored better than us. Like we were around 7 to 8 CGPA. They had around 9 to 10. Okay, though even then they have told we don't want to ruin our CGPA. But we have decided, we have taken 2-3 extra people Okay, like we went around 5 people. We went to his course. We have told them we are very much interested in attending your course. Okay, so we are registering registering for this course with you. Then a week has gone. He has conducted classes. It was very good, like interactive sessions. It was so good. Okay, we have enjoyed. After that, he has conducted exams and little bit tricky things. Okay, like continuous exams, like question and answers. It is not like monologue. It is like dialogue. Okay, if he teaches something, he will ask, "Did you understand these things like this?" Okay, he will ask us to repeat. Several things has happened. Among 5, 2 have dropped in between. In between means first after 2 weeks they have dropped the course. Only 3 were there. Okay, among 3, like one guy, Shantanu Singh, he is my close friend. Okay, he joined in Biocon after his MTech, and he went to Iceland. He is working in Alvotech. And that second girl, okay, she has recently she has completed her PhD in short duration, 3 to 4 years. The thing is, she has scored ranking of 30 in CSIR. She is a top, brilliant girl, and she has joined in Agilent. She has done her PhD in LCMS. Her complete PhD was in LCMS only, and I have joined in IIT Guwahati for my PhD. So something like challenging things, like, yeah, he is a tough person. Okay, when you take this kind of tough things, you can move on in your life and you can learn lots of things in your life. So this kind of things, why I am saying, try to face the challenges, face the fear. Okay, if you face these kind of people only, you can face your life. Okay, for example, one of the company in our India, they claim we are selling Ayurvedic products for treating blood pressure, for treating diabetics. Like that, they have sold their products in the market for a long time. One of a person like him only has filed a case against that company, stating that their product is not doing anything. They are making false allegations. I am not bringing that company's name. Everybody knows. I think so. Okay. It is topmost brand, and Supreme Court has sued them like anything. Yeah, because none of the doctors they will do. Most of them they won't do. Only very few people. The person who has filed case, he is also from Kerala. That much I know. Okay, that is the thing. You have to face the thing, and we need lots of people like him. Okay, every kind of people are needed, but like him also we need somebody who has to challenge the system, somebody who has to challenge what is basics which are wrong, like textbook. What he has taught us during the course is, when you are reading the textbook, they also would have made some mistakes. Okay, don't follow the mistakes like that. Try to correct it and try to verify what is wrong and try to fix it. That's what he has told. Like, find the gaps and fill the gaps. Okay, then one fine day when I was doing PhD, he has called me, Surjith, these are the works I've been doing. So could you, whether you can do these experiments for us? That's the thing. He has told. I didn't do anything, like most of the thing, majority of the thing, Abhinav, Andrew, what they have done, it's huge, humongous work, huge work, what sir has done also. But I have done couple of experiments. What I have done, that I will be telling you. 2 experiments I have done. Experiment 1 is to prove ROS is involved in ATP production. Okay, that is using luminescence assay. Experiment 2 to prove cyanide inhibits ROS, which reduces ATP production. That is using HPLC. Okay, when we come to luminescence assay, first thing is firefly. Okay, like how firefly glows in the night time. Okay, this is like in south they call it as a mini puchi. Night time you can see it glow. Okay, and second is cells activity. When cells are active, more ATP will be produced. Okay, this fireflies glow because of luciferase ATP. This reaction I will be explaining you. Okay, okay, this is luciferin. It's a substrate. Okay, this is luciferase. Luciferase is an enzyme which converts luciferin to oxyluciferin in presence of magnesium and oxygen. Okay. For example, luciferin is a substrate. In presence of luciferase, it is converted into oxyluciferin and it produces light. Okay, in this ATP is involved. This here ATP will be there. ATP will be converted into AMP and phosphate. This is the main thing. The thing is, this luminescence, when they are emitting light, you can measure using luminometer. Okay, it is also like UV spec. For example, in UV spec, you will be having a cuvette in which you will pass a light. Here detector will measure how much amount of light is coming out. Okay, it will give you observance value. Similarly, here in luminometer, you will put some substance which emits light. Okay, here sensor will be there, like lux meters will be there. It will measure amount of lights emitted. Okay, through that you can measure. You will come to know how much amount of light is emitted. Okay, this is luminometer. It is manufactured by Promag. Okay, first thing is background. Background is called, when you do UV spec, you will have blank readings. For example, you take water. Water in cuvette, if you put, you will have some observance that is like 0.01 or 0.15 like that. Even if you put nutrient broth also, it will give some values. For example, water can give 0.1, sometime broth, any broth can give 0.152, even 0.20. Okay, it varies, like basic values that you will be removing. For example, auto zero you will do. Okay, that means you will make it zero. That is the thing. Similarly, here, for example, distilled water in luminometer, it was giving 937 luminescence. Okay, like superoxide. Superoxide is potassium oxide, potassium superoxide. You dissolve in DMN also you can dissolve in water. It was giving 118. And only ADP dissolved in water, it was giving 758 superoxide plus phosphate. Okay, that was giving 936. Okay, these are all basics. When you put them in the luminometer, it will give. What was the experiment we have done was, ADP plus phosphate in presence of superoxide. Okay, here we can get ADP directly. It's a chemical component. We can get phosphate also. It is a source, like sodium phosphate, like that thing. Superoxide, KO2. Okay, so sorry, potassium superoxide. Here, if this superoxide is there, they will convert ADP to ATP. This is the main experiment we have done. So this is blank. Okay, positive control. Here ATP. When you put ATP with luciferase, they give this much amount of luminescence. That means 1825. When you put this is higher concentration. This is 0.01 micromole. This is 1 micromole. So intensity increases. It is around 21,000. Okay. Similarly, when you put ADP plus phosphate plus 2 mul of KO2, it was giving this much amount. That means 11,000 something, 11,421 lux. It was giving luminescence. Okay, when concentration of superoxide was increased, further this intensity has increased. Okay, after that, this experiment, this was slowly we were adding 2 plus 2 mul additionally, like sequentially we were adding, like it was like 5 shots. So this has given 18236 luminescence. Similarly, when concentration was increased in sequential manner, 10 mul, it was giving 33100. It signifies this experiment signifies that superoxide is involved in ATP production. Here nothing was there except superoxide, ADP and phosphate. Okay, this here we have proved that superoxide is producing ATP. Next, in this method you can say you can quantify only the amount of ATP, but you can't quantify the amount of ADP. So we have gone for HPLC also. In this experiment we want to prove that cyanide inhibits the superoxide. Here we have added cyanide. Okay, here reaction 5 we have added cyanide. I will tell you how it happens. First we have done this in HPLC. HPLC 2 modes are there, gradient and isocratic mode. In isocratic mode you will be running in same same flow rate, but percentage of solvent you will be fixed from initial to final. But whereas in gradient these things will vary, like flow rate also, the percentage of the solvents also will vary. So here we have used gradient mode, like these are the flow rates. Here when you put ATP and ADP in HPLC, you will get the peaks like this. This is ATP peak. They will be coming between 5.2 to 7.5. This is ATP and ADP. For different concentration we have got the plot. Okay, this is the standard plot. Here standard plot was 0.9, R square was 0.99 something like. Okay, here positive control we have added 200 micromole of ADP. Here we have got the peak for 213. Okay, using this standard plot we have got 213. That means for 200 we were getting 213. That is a deviation. It tends to happen. That is a machinery deviation, or even handling, anything can happen for everybody. That's why experiments are done thrice to get the deviation. Negative control, here we have added only phosphate and superoxide. Here intensity was not there. Another negative control was only ADP 200. Here we were getting 211. Test 1, this is 200 micromole of ADP plus phosphate. Here we have got 202. Okay, here superoxide has produced ATP, and fifth one was with cyanide. Here ATP was 2.1. Sorry, this is ADP 202 was ADP 2.1 was the ATP. That fifth one, here when we add the cyanide, the ATP level was reduced from 2.1 to 0.7. Okay, this is the two basic experiment I have done. Through this we have proved that using luminescence assay we have proved that ATP can be produced using superoxide, and second, using cyanide we can say that cyanide inhibits the superoxide, so ATP production also reduces. That is how cyanide works in the human body. Next thing is few things I want to say about AI modeling. AI and modeling. Why AI and modeling is done? The thing is, like, I work in a precision fermentation company. I know one company in Singapore. They have produced meat protein. Okay, like hemi protein consists of iron containing protein, which tastes like it is a plant protein which tastes like meat. What they have done is they have taken the meat protein, they have analyzed using AI tools, okay, to find which plant protein is close to this meat protein, and they have found one protein, and they have extracted from the plant, and they have made a food material, food like meat kind of thing, natural meat, which is almost tasting equivalent to animal meat. Okay, this is how AI is applied in real time in food industry also, biotech industry also, and lots of things, like even before, for example, genetic engineers, before going for like recombination technology or anything, for finding the combination, which promoter is suitable for expressing specific protein. Everything they are analyzing using AI only, like AI and modeling also. They will do modeling and they will try to get best results. For example, score which is closer to 100. They will like models will do like around 100 experiments. Among 100 experiments, first 3 will be chosen based on the scores. That thing will be carried out as an experiment. When you do this 100 experiment, you will be losing out lots of substance like financial troubles are there. So first you do the modeling, then you will choose the which experiments to do, then you will go for the real time in vitro experiment. Then homeopathy kind of thing, like what I have analyzed is like picomolar concentration. It is there. It is not even nano. It is a picomolar. Very less amount of substance are there in the. They are climbing to be very very less. Even homeopathy, like any kind of experiment, it has to be that. Sorry, any kind of medicines, it has to be approved. Okay, it has to be approved. On top of that, you have to read the papers, like how this specific substance works to treat a disease, to cure a disease. Proper evidence you try to find. Don't believe anybody blankly saying this is going to cure. Okay, scientific community, peer reviewed papers, that paper also will be challenged in several places. Okay, experimental data from